Good morning, everyone, or perhaps I should say good evening or good the middle of the night. My name is Sara James, and it is my real pleasure to welcome all of you to the GETA Genetic Epilepsy 2020 Summit. And we are thrilled that you've joined us, and we are especially thrilled for all of you who've joined us from around the world. I was looking at my phone just a moment ago, as we all do all the time, and I was checking the time zones, and I was thinking about the effort that some of you have made to be here and to be with us, because we have folks joining us from all across Australia, but also from the United States. And it's 4 o'clock in the afternoon in Los Angeles and 7 o'clock at night in Washington, D.C. So thanks for being here with us. We also, though, have people who are joining us from London. So a very good early morning to you. It's just after midnight there. And from Yemen. And my phone tells me that it's 2 o'clock in the morning where you are. Also from Brazil, from Spain, from Germany, from China. And we can't tell you how much it means that you are here, that you've found time in the middle of your busy lives, in the middle of the night, to join us and to be part of our summit. But we know why you're here. And we know that you're here because genetic epilepsy doesn't stop. It doesn't start at 7:01 in the morning and end at 7:45. It's not something that you can schedule like you can schedule a Zoom call. It affects everything in your life, everything in all of our lives. And I think that's why we all made time for this today. There are a few little differences. We had planned to have all of us meet together. And if there's one disappointment that I have, it's that I can't say hi in person to all of you and get a chance to meet you and to hear your stories. That's the downside. The plus side of having this be a Zoom conference call is that more than 350 of you have found the time to join us. And that's a bigger number than we've ever had. So credit to you and thanks to you. And we really appreciate that you're joining us on Zoom. So where are we, by the way? Well, we are in Melbourne, Australia. And this is my office. Well, it's my husband's in my office. And I say that because really what's interesting about GETA is this got started by 3 husband and wife teams. So this is our place here in Melbourne. But if you give me just a moment, I'm going to bring in some other people. We're going to try to observe good social distance, but we're only one tiny part. Andrew and I are only one tiny part of GETA. So let's have a look. And I'm going to introduce you to 2 other couples who are part of us. This is Danielle and Danny Williams. Say hi, guys. Hi. They were busy moving furniture just about a minute ago. And also we have Chris Pierce and David Cunnington. Say hi, everybody. And we're saying hi to all of you. This is thrilling. Do you guys want to say anything while we're just standing here?
Just welcome and thanks for dialing in at whatever time of day or night it is.
Yeah, great to have everyone here.
It really is. It's thrilling. And, you know, everybody does so many things. Don't you go anywhere, Danielle. I can see you. But Chris and David put together the incredible website. And if you haven't had a chance to be on that website yet, please take a look. Chris does a great podcast as well. So you're going to want to have a listen to that. So have a look at what they're all doing. And while you're on that website, and also you can find this on Facebook, and you can find this on Twitter, and you can find this on the email when you signed up for us. Danielle has put together something on SurveyMonkey. And that's going to be a form that we really want you to fill out, because it's going to give us an opportunity for us to know who you are. And I can tell you that 88 of you have already filled it out. So I'm going to tell you a little bit about you. But before I do that, I want to say thanks to my friends.
Thanks,
I won't make you stand there, but thank you very much. I'm only a teeny part of this, you guys. All right, so now let me tell you about you. So let me start this way. Around the world, 1 in 26 people are likely to be diagnosed with epilepsy during the course of their lives. 1 in 26. That is an enormous number. And not only is that a significant number in and of itself, but a substantial number of the people who are diagnosed with epilepsy every year are going to be diagnosed with genetic epilepsy. And that's a fascinating thing to consider, because of course it wasn't so very many years ago that we didn't know such a thing existed. And I am delighted to be able to tell you that during our call today, during our Zoom meeting today, you are going to have an opportunity to hear from 2 people who helped discover that very first gene for genetic epilepsy. But all of you are here because in one way or another, your life intersects with genetic epilepsy. Let's talk about just some of the genetic epilepsies that you represent. My family, ours is KCNQ2. So a shout out to our friends at KCNQ2 Cure and for all of our families with KCNQ2 who join us. But we're just a tiny, tiny part of this. What about Dravet? We know that of the people who filled out our survey, the largest number so far have Dravet. And of course, that was one of the very first diagnosed. Let me tell you about some others. SCN2A. SCN2A is here. Also, LGS, SCN8A, KCNA2, SYNGAP, and CDKL5. Now, our other two families represent SCN2A, that's Chris and David, and also SYNGAP, that's Danielle and Danny. But I want you to know something. No matter who you are, and no matter what genetic misfire you're representing, we want you to know that you're just as important to us because this is our community. We are all in it together. I sometimes think of it as an alphabet soup. It's as if you kind of put your spoon into that soup and you pull up a set of letters and numbers. And in and of themselves, when you first get that really stomach-wrenching diagnosis and somebody gives these letters and numbers to you, and you're looking at your beautiful newborn baby, and you're saying, that's not a representation of who they are. They have a name, they have beautiful eyes and sweet little hands, and they're a precious child. They're not those letters and numbers. So no matter who you are, and no matter what the letters and numbers are in your diagnosis, we're really glad you're here because you are all precious to us. And there are many, many things that all of us share, regardless of what those letters and numbers might be. And that's really why it is important if you can take the time to fill out that survey on SurveyMonkey. By the way, what did we do before SurveyMonkey? I mean, I feel like I fill out so many of those. So please take the time to do that and tell us where you're from. That's how I know where so many of you are joining us from. And if I haven't mentioned your country, or by the way, your state, let me know. And that includes your state in the United States or your state or territory here in Australia, or wherever you are in the world. Just let us know. We want to put some context and detail and depth to our knowledge of our community, and that's incredibly important to do. One other thing I should mention before we get started today, and that is that we'd like you, if you can, to tweet, to write us, to ask us your questions. Maybe we'll have an opportunity to talk to our experts about that today. And if not, we'll follow up. That's what we like to do. You can tell us if there are things you like, and you, by the way, can also tell us if there are things you don't. So feel free. We want you to communicate with us, and Zoom makes it, in some ways, a little bit easier and more friendly. Now, I mentioned our experts, and I don't want to keep talking without getting to our first expert, particularly for some of you who are far, far away and are staying up in the middle of the night. I'd like to first introduce you to Sam Berkovic. Now, we're lucky to have Sam here in Melbourne. And, you know, I always feel a little funny when I talk about these esteemed scientists and I call them Sam, as opposed to Professor Berkovic or Dr. Berkovic. But may I just say, that is the style here in Australia. It's a very casual country, and I confess that I have gotten used to it. It's also true that these scientists, over time, we've gotten to know them well, and they have become our friends. But I don't want you to be confused and think that just because I call Sam by his first name, I show him any less regard, because that could not be further from the truth. Sam has many, many honors, including that he was elected a Fellow of the Royal Society in 2007. And another of our speakers, Ingrid Scheffer, was just explaining to me what a very, very big deal that is for scientists. And she explained that Sam is walking in the footsteps of Einstein and Newton. So why is he getting those accolades? What is that attention along with his many other honors? What is it for? Quite simply, it's because of why we're all here. It wasn't so many years ago, back before the 1990s, where scientists around the world did not believe that genetic epilepsy existed. It's always interesting to think about those times and to recall something that we take for granted and that we now know in some degree of granular detail, and recognize that not only was it not accepted, it was dismissed as a fallacy. That work, not only to discover the gene, but to combat that misconception, that is the work of Sam Berkovic. Back in 1995, he, along with some molecular genetic collaborators in Adelaide and Germany, and also with Ingrid Scheffer, Professor Scheffer, from whom you'll hear later, he discovered that there was indeed such a thing as genetic epilepsy and discovered the first gene in 1995 for genetic epilepsy. That changed the playing field for all of us. And in the years since, hundreds of people have been diagnosed, hundreds of genes have been discovered for genetic epilepsy. So now, giving you that small background, I hope you'll allow me to introduce you to Professor Sam Berkovic, who will give us an overview and history of genetic epilepsy. Sam?
Thank you very much, Sarah, and thank you to yourself and Danny and Chris and all of GETA for organising this. I'm really pleased to come and talk to you, and I must say I've been amazed watching GETA from the sidelines, so to speak, as to how much you've achieved. I've been asked to give an overview of genetics worldwide, and once I get my slides moving, yes, so what I'm going to address is the history of epilepsy genetics, which is actually quite ugly. And when you understand it, or when it sort of explains what's gone on in the last few decades and what is still going on. I'm then going to address give a helicopter view of the last 30 years of epilepsy genetics, and then specifically address the relevance of genetics to the developmental and epileptic encephalopathies, which I'll refer to as the DEEs, what we've learned about it, what are the surprises, and where are we going. So, in fact, knowledge about the inheritance of epilepsy has been there since the beginning. And the great religions took a pretty dim view of this. For example, in the Talmud, it was advised against taking a wife from an epileptic or leprous family. And this is sort of somewhat amusing, as of course leprosy is an infectious disease, albeit one with some genetic predisposition. But it was believed that both were inherited. Hippocrates, the great Greek physician, recognised inheritance in epilepsy, and he was very positive about it and saw it as a physiological disease, and he called it the sacred disease, really as a counterpoint to the fact that it wasn't mystical or whatever, but used that term to try and highlight that. But unfortunately, that was pretty ineffective, and epilepsy was still shrouded in a lot of stigma. In medieval and Renaissance times, epilepsy was widely accepted as hereditary, and there was a lot of stigma. And this was reflected even in the relatively modern era, where there were restrictive laws on marriage, even in seemingly advanced countries like Sweden, and even in the United States. A number of states had legislation against marriage because of the genetic factor, and the last one was repealed only in 1980. And sadly, the stigma associated with genetics is still an issue in some non-first world countries, and even in some first world countries, one might add. And indeed, as Sarah indicated, the medical community in the last 50 or so years regarded genetics in epilepsy as something pretty minor. And here's a major report from the National Institute of Health, 1978, saying that inheritance plays a very minor part in epilepsy, despite the widespread misconception that epilepsy is inherited. Now, in fact, that is now known not to be a misconception. They acknowledged that there were some unusual syndromes that were inherited where there was epilepsy, and there was an issue of susceptibility, which is also true. But again, the issue of genetics was downplayed. And this puzzled me when I started getting into this field about 30 to 35 years ago. And what I'm going to present to you is opinion and not data. I think there are two very important reasons why there's sort of been this ambivalent view about the genetics of epilepsy. So first of all, there is good data, and there was good data 50 odd years ago. And one of the leaders in this was an American neurologist called William Lennox, and he studied twins, and twins are a really good way to show whether things are inherited. And here are a pair of attractive young girls with childhood absence epilepsy, a very mild and self-limited form of epilepsy. And you can see that they're identical, and they have identical EEGs. These are the discharges associated with absence. And Lennox was extremely impressed by this. And when he saw a twin with epilepsy, nearly always the co-twin had it as well with this form. And this allowed him to start to think about the fact that genetic factors were important. Yeah, twins are marvellous subjects. They're interested in the fact that they're twins, and they're very, very useful for clinical science. And we actually managed to follow up this pair many, many years later. You can see they still like being twins, and they grew up to be very healthy and productive women. They had a bunch of children and grandchildren, and none of them had epilepsy. So this illustrates the paradox that in twins that have exactly the same genetic composition, the epilepsy frequently is seen in both, whereas it's much less frequent in their other relatives who are not twins. And we'll come back to the explanation of that in a moment. So why, in the face of this evidence, was the genetics downplayed? Well, as I said, I think there are two reasons. One is horrible, and that is the eugenics movement, which started around the turn of the 20th century. And there was a move allegedly driven by altruism that we would improve society by getting rid of children who were not perfect, if you will. And this was led by some seemingly esteemed people, since all discredited. But as you know, it led to the horrors of Nazi Germany. And this stain on the genetics of epilepsy really persisted through the second half of the 20th century and really made genetics something you almost couldn't talk about. And that was sort of the environment that one faced when we were starting to do this work years ago. The second is not a malevolent reason, but it's sort of a scientific reason, and it's the way we learnt about genetics. Those of you that did a bit of genetics in high school remember the story about Mendel's peas, the Austrian monk who figured out the laws of heredity by growing peas. And those laws still stand. And this started to get applied to human diseases about 100 years ago. And one of my heroes is this guy called Archibald Garrod, who long before the discovery of DNA figured out some hereditary diseases and called them inborn errors of metabolism. In other words, something that was hereditary that led to a change in the way the body worked. And he described a number of rare diseases. And then in the middle of the last century, molecular biology came along, and we started to understand how we went from genes to proteins and then to diseases. And there were quite a number of diseases described where a single interference with a single protein led to a particular disease, and so-called one gene, one protein, one disease. And these are sort of Mendelian disorders, which are true and are rare, and there are many of them, and they're important, but they don't represent the majority of inherited diseases. So, in short, the answer to the paradox is that epilepsies have a major inherited component, as I'll show you, but few epilepsies show simple or Mendelian inheritance where things go through families like a golden thread, like Garrod described. That's not the rule for most patients with epilepsy. Instead, they have so-called complex or polygenic inheritance where many genes contribute. And this is a solution to the paradox of the twins, where the twins often are both affected, yet their first degree relatives, their children and their more distant relatives, are much less frequently affected. And this is what you see in complex or polygenic inheritance. Thirdly, and relevant to the DEEs, some epilepsies have what we call hidden inheritance, where there is in fact a genetic cause, but there's no family history. And this is often due to de novo inheritance or de novo mutation, a new mutation. And here you in fact get a genetic disorder without affected relatives. So these are the reasons, I think, that why there's been this confusion over the last millennia about it. Now, cracking the actual molecular basis of the epilepsies is also a very remarkable story. You could argue it began with Watson and Crick in 1953, who figured out the structure of DNA. And the DNA is made up of what we call base pairs, and we have 3 billion of them, an enormous number. And to find a genetic cause amongst 3 billion is a seemingly impossible task, but that is something that has been achieved. We have 20,000 genes or so. We have 3 billion base pairs. In the 1980s, the ability to map, genetic mapping began. The first gene discovered through this technique with no prior hypothesis as to what it might be was discovered in 1989 after an enormous amount of work. 10, 15 years of work by many, many laboratories found the gene for cystic fibrosis. That task now would almost occur just in a couple of days with our new technology. And this was enabled by the Human Genome Project, which began in the 1990s in the United States. It was regarded as sort of the moonshot for biology. There was a lot of criticism about it. Everybody thought too much money was being spent and wasn't going to get anywhere. And then in 2001, it was regarded as completed, although it wasn't strictly completed, but it was a good start. And that cost $3 billion. And Bill Clinton announced that in 2001, and there was an interesting side story about that, with a big sort of race between the government-funded project headed by a guy called Francis Collins, who continues to be very, very prominent in American medicine, a very brilliant guy, and a maverick called Craig Venter, who led a private consortium. And they basically got there together. It was a draw, and they probably spurred each other along. But that was the Human Genome Project, $3 billion to sequence the human genome. Now, the spectacular thing that's happened that's enabled us to apply this to human diseases has been the improvements in technology. And this is a graph that shows the cost of sequencing one human genome. To do the first one was $3 billion, and once it was done, it was estimated it would cost about $100 million to do the second one. And this is a logarithmic scale. Today, we do it for $1,000, which is something that's tractable, it's affordable by the medical system, it's still relatively expensive, but this has been brought about by absolutely remarkable advances in technology. We all learn, or we all know about technology and the rapid changes by the fact that every time we buy a computer or a phone, the memory of it improves and the capabilities of it improves. And this has actually been described by what's called Moore's Law, which says that the computing ability doubles every 18 months. So what this shows is that the advances in genome technology have far outstripped Moore's Law. This field's been moving much, much faster than computer science in terms of the ability to do things, at least on a cost basis. So this has really driven enormous advances in medicine. So that's sort of the overall background. What about what's happening in epilepsy? Well, first of all, I'm a clinician, and you'll hear talks from a number of other clinicians this morning, Lynette and Ingrid, and it all starts with the patient. So what's the evidence that actually epilepsy is genetic? Because if I would ask some of my neurological colleagues, both paediatric and adult, how often do they see patients with genetic epilepsy? Well, how often do you see a family history? Oh, well, we see it, but kind of not that often. And that's sort of embedded in that view that I showed you in that 1978 slide, that the importance of genetics wasn't all that great. So the actual data is that when you look at particular types of epilepsy, we can measure what's called the familial aggregation, the likelihood that first degree relatives will be affected. And if there's no genetic effect, this number should be 1. And what this shows that is in generalized epilepsy, there is a large genetic effect. This number is 8. And that implies that genetics is very important. But it's also true in focal epilepsy, where it's well above 1, but still nowhere near generalised epilepsy. And in febrile seizures, it's somewhere in between. Epileptic encephalopathies doesn't appear on this because you don't, for the most part, see familial aggregation, or only very rarely, because now we know that this is a de novo disease for the most part. We also learnt about this from twins, and I did a lot of my early work in twins, which sort of gave me the ideas about where we might look for genes. And what we look for here is the concordance. If one twin has it, how likely is the other twin to have it? And when this figure is 1, then that means that the whole story is genetic. And you can see with generalised epilepsy, the figure is 0.8, which is about as high as it gets in many diseases. So this means that generalised epilepsy is a very, very heritable disorder. Similarly, in focal epilepsy, it's much higher than previously thought. These techniques, however, don't get you to the genes. And that happens when you get, this happened in the past when one was able to study very large families. And this was the work that Ingrid and I began doing in the early 1990s, where we collected large families and tried to figure out where the genes were with molecular collaborators by the techniques available at the time. The Human Genome Project was still well off in the distance, and one couldn't use that. So this was work from Ingrid's thesis, where we initially started off with 2 very small families with what we recognise as an unusual form of nocturnal frontal lobe epilepsy. You can't find the genes in such small families, or you couldn't with the technologies available in the 1990s, but you can if the family's larger, but nobody had really identified such families. So the work that Ingrid did was basically go and ask everybody about epilepsy in the family and build a big family tree. And when she did that, it turned out to be this. These 2 families that were not known to each other were actually related, and this was one enormous family spread across southern Australia. And the information from this family and collecting their blood samples and analysing them enabled our collaborators to map this to chromosome 20. And then we were lucky at the time, not all the genes were known, but the gene that turned out to be the one in this family was known, and we identified it as the nicotinic receptor of the nicotinic receptor, which responds to acetylcholine, one of the chemicals that's particularly prevalent in the front part of the brain. And this was the first gene for epilepsy, and was the paradigm for what we call the channelopathies. In other words, epilepsy is now known to be, at least in part, a disease of channels, which is the proteins that lead to communication inside and outside the cell. And in terms of nerve cells, determine how excitable they are. And that is, of course, very sort of a very sensible mechanism for the way you might get a hyperexcitable disease like epilepsy. So this is then a time chart of the scale of the speed of gene discovery. Here was the first one, this nicotinic gene, and then there were a bunch of other genes discovered in large families. Most turned out to be channels, but not all of them. And then there was a big kick up in the gene discovery, and this goes higher and higher. This scale's now a lot older. Most of the genes here, some of you are quick will recognise some of them. Most of them are genes for DEEs, and this was enabled by the new technology of the Human Genome Project. So what we learnt was through the Human Genome Project's technology that there were many new genes for epilepsy, many single gene epilepsies, particularly in the DEEs, as we'll get to. But we're also very interested in genes with polygenic inheritance, the ones represented by those twins particularly, where the evidence was that the inheritance was complex and not determined by a single gene. And there are two basic hypotheses for these. One is that common diseases are due to a small number of what we call rare variants, things that occur in all of us, but on their own don't cause disease. And these increase the risk of having disease by of the order of sevenfold or more. And they occur quite rarely in us, and they're detectable by basically sequencing everything by whole exome or whole genome sequencing, which is now possible. The other hypothesis is that common diseases like the common forms of epilepsy are due to numerous common variants. And these we all have tens of thousands of these. They are subtle changes in our DNA that on their own do absolutely nothing, but they're studied by what are called genome-wide association studies, or GWASs. And all these techniques need very large cohorts to crack the answer, because it's clear that there is no sort of single gene for epilepsy. There are many, many genes. We already know of hundreds. So no single lab can do this, and collaboration in the last 10 or 15 years has really been the key. There are large international consortia, the ILE Consortium on Complex Epilepsies. I founded that about 10 years ago now. There are some now large collaborations driven by the NIH, Epi4K, which we were heavily involved with and now completed, where we sequenced 4,000 epilepsy genomes. A large European study where we also collaborate with. And then Epi25 was a project funded by the NIH, sequencing 25,000 subjects with epilepsy, and it's now in its last year. Also, this has been greatly aided by the internet, groups communicating with one another, techniques or programs called Gene Matcher, where if you find something in your lab, you can very quickly figure out if another lab has found it. And also, very importantly, the power of patient and parent groups to actually link together, not only to support each other, but really to help drive research and bring patients with similar disorders together to the attention of researchers. So this has really been a seismic shift in the way science works, and has really generated an enormous amount of data. Now, the short answer, and I won't go through all the details because the answers still aren't in, but it appears that the common epilepsies are due to a mixture of a rare variant. And here's an Epi4K study that shows these genes indicated here are the ones that are important in focal epilepsy. And we're starting to pull out genes that are important for the so-called oligogenic architecture of the epilepsies, where a relatively small number of genes may contribute. And this is a genome-wide association study where you get these plots, and the higher the plot is called a Manhattan plot because it looks like a skyline, Manhattan skyline. The plots, the ones that go above the red line, are significant, and we've now discovered a whole bunch of genes, and more coming all the time, that are contributing to epilepsy. This unfortunately doesn't yet lead to direct genetic testing because it's each individual gene doesn't contribute all that much, but we're learning how to use this. And one of the exciting new things, and this is all very new, is the utilisation of so-called polygenic risk scores. And a good way to illustrate this is that we all know that height is something that is inherited. If you've got tall parents, you're likely to be tall. Here's a young man who's extraordinarily tall, nearly 2.3 metres, a well-known basketballer called Sean Bradley. He's healthy. He's a very good basketballer. And why is he so tall? Well, this has been answered by looking at the so-called polygenic risk score for height. And here is the polygenic risk score of a bunch of random individuals, and it's what we call a normal distribution, a bell-shaped curve. And Sean's out here. So he's tall, not because he's got a single mutation affecting some particular gene, but because he's got an aggregation of a whole lot of very minor effects that come together and make him so tall. And we're now using that strategy in diseases like epilepsy to try and understand this, and you're going to hear about polygenic risk scores more and more in coming years as we apply it to clinical diagnosis. So just to circle back to the DEEs, what have we learnt? So 30 years ago, a child with intellectual disability and epilepsy, the diagnosis was just that. Thank you very much, Mr. and Mrs. Jones. Your child's got epilepsy and intellectual disability. We basically can't do anything. It used to be a diagnosis, and I put that in inverted commas because that would no longer be acceptable. And it was often attributed wrongly to a perinatal injury, a vaccination, some other trivial event, or probably the truth was we just didn't know. Now we know that the DEs have a small number of obvious causes. Severe perinatal injury can cause it, and there are some severe early infections that can cause it. But most of the rest are genetic. It's highly heterogeneous. Most of the recognised forms occur de novo, that is, there's a new mutation. There are some recognisable clinical patterns, and there's much better conventional treatment. Knowing a diagnosis helps, even if you can't directly affect the cause. What were the surprises? Well, I certainly didn't expect, and I don't think anybody expected, the degree of heterogeneity that we've now got hundreds of causes of the DEs. It should have been obvious, but it wasn't. But the importance of de novo mutations, that explains why, for the most part, there's only a single affected child and there's no family history. We've learnt about some unusual genetic mechanisms, and genetics as a field has benefited from the intensive study of the DEs, some that occur by unusual X-linked mechanisms. We've also learnt, and this is very recent work from Ingrid and Heather Mefford, the importance of mosaicism, where instead of a truly novel mutation in a sperm or an egg, there's a very low level in the parent, and that's important to determine recurrence risk. We've learned from the genes that there are a diverse number of functional effects that's really informing the way we think about these disorders from a biological viewpoint. And at least in the laboratory, the speed of new treatments where we can treat animal models of these disorders has been a lot faster than I anticipated, and that's a really wonderful thing. Where are we going? Well, we hope we can solve all the cases. One thing I've learned from coming to the GETA meetings, that even though you don't want to be known by letters and numbers, there's only one thing worse than having a letter and a number is not having a letter and number. In other words, where there's no diagnosis. And we hope that we can solve all of them and get to precision diagnosis. And exactly how many are solved is a bit of a guesswork, but probably 60 to 70%, maybe higher. We've underestimated the number of recessive disorders because they're actually hard to detect, but we're learning how to detect them. There are areas in the genome that are difficult to sequence, and we're getting better at doing that. So we're discovering hitherto undiagnosed cases with that. And we're learning more now how to use so-called whole genome sequencing as opposed to exome sequencing, where the bits in between the genes are sequenced, and that's going to yield up a lot of secrets. And in terms of understanding the disorders, the neurobiology and the treatment, they're real advances in areas that were previously regarded as hopeless. And you'll hear a lot more about that from Ingrid and from Steve later in the morning. So to summarise, there's an ugly early history of epilepsy genetics that held back progress. Stigma is still an issue in some places. There have been remarkable advances in the last 30 years that have been enabled by technology, specifically the Human Genome Project, but also by the realisation that the only way to work in this is in a collaborative way, both at the level of countries, individual labs, and parent and patient groups. For the DEEs, genetics has completely changed our understanding of all the epilepsies now. This is the group that we understand the best at the genetic level. So that it's gone from being sort of a Cinderella of epilepsy to the one that we know the most about. And there is real hope for new treatments, as you will hear. And again, this is very much a collaborative game. You've probably seen similar slides to this before. But our work is only possible by working together within our group. And I've listed some, but not all of the people here, but obviously particularly Ingrid and Steve Petrou for the functional work I want to highlight. So thank you very much for listening to me.
Thank you so much, Sam. I'm really appreciative. It's a lot of interesting information to digest, and I do have a couple of questions that are already cropping up from many of our listeners who are online, and we have a few more countries to mention as well. I think I've mentioned many, but I'm just going to go through quickly. The U.S., and among the U.S., we have New York, Philadelphia, Pennsylvania, we have Florida, we have the U.K., we have Australia. I think most states and territories are represented: New Zealand, Canada, Spain, Germany, Brazil, Yemen, Egypt, Iran, and China's not on this list, but I know we've got somebody from China as well. So you've got a lot of people listening to you. And Sam, I'll ask you a couple that are specific and then a few more that are a little bit more general. One of our first questions comes from the UK, and it's very specific. Any information on SCN1B? B for boy. Is that one of the ones that you've been instrumental in diagnosing, Sam?
Yeah, that was, if I remember correctly, gene number 2. So we initially described this in a condition called genetic epilepsy with febrile seizures plus in a very large family from northern Tasmania. And for the most part, variants in SCN1B cause a pretty mild disorder. Some of the children in that family were more severely affected, but it's now very rarely been described in much more severe children, particularly if you get 2 abnormalities of that gene. But again, you need to look at the details in each individual case to give more specific information there.
And you may say that about our next question, but I'll go ahead and ask this as well. And this is related to KCNQ2. And the question is regarding, and I actually think this is relevant because it's true with so many of the DEEs. The reality is that the seizure disorder is only a tiny portion of what families are dealing with. So this question is about a 5-year-old with KCNQ2 who has just been diagnosed with ADHD. And a question as to whether or not medicines for ADHD are safe and effective for KCNQ2 children and beneficial. And I think basically a question about what to do, and is a medical route, either ADHD or many people are looking at CBD, or a behavioral route, the right way to go. So I'm just wondering if you can speak maybe a little bit specifically, but generalize, because so many parents are in the same predicament of looking at not just the seizures, but all the attendant challenges that come with genetic epilepsy.
Yeah, so I perhaps should have emphasised that more, that obviously the DEEs are much more than just seizures, that there are a large number of comorbidities, and understanding the biology of that's actually very interesting and very complicated and still not very well understood. So with the epilepsy, of course, and the DEEs comes intellectual impairment, often behavioural abnormalities, sometimes very severe behavioural abnormalities, and on that spectrum, ADD. Now, ADD per se is a very common thing. So when ADD occurs in a child with a DEE, it may not necessarily be due to that, but it probably is. And I think for the most part, and Lynette or Ingrid, who I'm not a paediatric neurologist, Lynette or Ingrid would be better at answering this, but at least my general view is that you treat those symptoms on their merits. There's no particular strategy or medicine that you should or shouldn't use to treat that particular symptom. But again, you're not treating epilepsy or ADD, you're treating the whole child, and it has to be taken into account of the whole situation.
All righty. And we know that in a forum like this, obviously, we're not expecting a diagnosis or prescription for individual children, but I think it's relevant, and we appreciate you giving kind of this general guidance, because again, these are so many things our community, faces. Another question that's specific, and then we'll move to a few more general. How common is it to find multiple mutations in a single gene, for example, SCN1A? And this is from Paula in Canada.
So that's a very good question. And the answer is that's the usual story. And I presume the question is in different children with this. For example, in Dravet syndrome, there are many, many mutations that occur in SCN1A. There are some that occur recurrently. And SCN1A is a very large gene. So not every change in SCN1A leads to Dravet. And that's one of the puzzles or the problems that the clinicians and clinical geneticists have, that you have a child with Dravet or possibly Dravet, and you do a test for SCN1A either directly or through a whole exome sequencing, and you find a change, then figuring out whether it's relevant to that child is not necessarily easy. If it's been described before, it is easy, so that's actually an advantage. But, the short answer to that important question is that, for the most part, many different mutations in the same gene can have a similar deleterious effect.
This is all fascinating, and I'm going to continue to ask a couple of more, and we want to encourage people. It's great for you to send in these questions because we're happy to answer them or to have them answered by our experts. That's one of the real abilities that we have in a forum like this. Sam, this question comes to you from Ingrid in Australia. When will we know all the genes in epilepsy and be able to diagnose this at birth? And I see you smiling. I suspect you know the Ingrid to whom I refer. Cheeky of her.
The analogy I like is to compare discovery in science to engineering. Engineering's a wonderful science, but if you want to know how long it's going to take to build a bridge across a big river, you can kind of figure it out because it's been done before or partly done before. And you can say we're going to finish this bridge in 5 years. When you ask the question, when are we going to finish an answer in biology, we don't know. We don't know how many layers there are on the onion. We keep peeling the onion. I was once asked how many, it was actually at the first Epi4K meeting in 2012, I think it was. I was once asked how many genes did I think there'd be for DEs, and I said rather flippantly about 100. And a few years ago, I was congratulated on sort of getting it right. Now, I was right about 4 years ago. I'm now wrong. So
I get it. I really do.
We just don't know.
There's an Australian expression, Sam, how long is a piece of string?
Yeah, exactly.
And I think it's similar to that. But again, it is a powerfully important question. I did an interview recently with a disinformation specialist who said that one of the hardest things for human beings is sitting with, we don't know. Mm-hmm. And it's an incredibly important part of science. So I recognize that it's hard to give an answer, but do you have a general sense of when it might be? Are we talking 1 year, 10 years, 100 years?
There's a paradox there because that lack of knowledge drives scientists, but it frustrates families. So,
true.
Look, we've learnt an awful lot. If you were to ask me, when we started this, around the late 1980s, whether we'd be where we are now, I would have said no. I think we've achieved more than one might have thought about. But look, I think it's going to be a lot more heterogeneous than we thought. There will probably be thousands of genes that are important for epilepsy.
All right, I have two more quick questions and I want to keep us on time. We're running a few minutes late for those who are paying close attention, but we'll catch up. Two more. One is how important, you mentioned the power of family groups, how important is global collaboration when it comes to this? And the second question, which I'm going to ask you at the same time, is we're living in the days of COVID. We're all really familiar with the worldwide efforts to find a vaccine for that pandemic. So I'm interested to know also if that spills over into other scientific endeavors, particularly ours, or if it's going to hinder efforts to continue research into genetic epilepsy.
Well, international collaboration is just absolutely important. There's a tension for the individual scientist where he or she, wants to make a discovery, sometimes for altruistic reasons, sometimes for selfish reasons, and sometimes just to keep their salary. That you've got to be leading, but we've all learnt that working together is just a much more effective way of doing that. And in COVID, I think for the most part, one can see a fair bit of international collaboration. I didn't quite, I think there are a couple of ways I could approach the second part of the question. Are you asking whether COVID's going to hinder the research into genetic epilepsies? Is that?
I guess, will the focus and the funding that necessarily will flow on to such an unbelievably important virus, and we've got to get a handle on it, will that take money away from genetic epilepsy research? Or are they kind of tandem tracks? And kind of separately, could it actually spur things on because there is this huge scientific endeavor to find a vaccine? Science is getting an enormous amount of attention. So I guess I could play this as an optimist or a pessimist.
You can,
but my opinion is less important than yours, Sam.
No, I don't know. I think I'm not sure. I suspect the reverse may be true. Look, we don't really know. Historically, when there has been a major push for something, it doesn't tend to rob other areas of all that much funding. It's usually manageable. On the other hand, if there's a long-lasting worldwide economic depression as a result of COVID, that will affect science. There'll be less money for everybody. I think that's the thing to be most afraid of. But look, the more attention science can get, and the more kudos science can get, I hope Tony Fauci comes out looking a lot better than Donald Trump in the long run, and he's the one we respect, and I think that will improve the respect for science.
Anthony Fauci, yes, the head of infectious diseases in the United States. For those who might not be familiar with the name that you're talking about so familiarly, he truly is the gold standard, and his opinions, his scientific knowledge grounded in those facts. He's a voice of reason, and we're very impressed by him, not just in the United States, speaking as an American and an Australian, but around the world. Sam Berkovic, thank you so much for your time and for answering questions. We really appreciate it.
Thank you very much. It's been a pleasure.
And as we wrap up with Sam, one of the things that we talked about with Sam was COVID-19 and the global pandemic. And as we come to our next guest, it occurs to me to mention that one aspect of that virus, of the coronavirus that we now call COVID-19, is that it has caught global attention because it can affect anyone, anywhere, at any time. And it occurs to me that there is a parallel here with genetic epilepsy, because if there is one thing we know about our community, it is that it is international and it does not discriminate, that anyone, these are de novo mutations, and anyone, anywhere can be affected. We have, as part of our program today, taken the liberty of calling on some parents to get their views on what it's like, parents and grandparents and researchers, to tell us what it's like to live with a child with a genetic epilepsy or to research in this field. And so the first family that I want to talk to is the Nowers family. And now we're going to hear from a KCNQ2 parent. Her name is Jennie Nowers. She herself is a doctor. Let's have a listen to her journey.
Hello everyone. Thank you to Geta for inviting me to be part of the first virtual Parents Conference. I first attended Geta last year when I was the vulnerable parent of a 4-month-old with newly diagnosed genetic epilepsy, and I cannot put into words just how much of an impact it had on me. I was lost, grieving, and broken. I had the honor of meeting some of the most incredible mums and dads who spread their angel wings around me from the first moment, which, incidentally, was as a tearful, strung-out mess in an unfamiliar city, standing in the foyer of the Florey Institute, waiting for a lab tour I didn't want to miss, but I had forgotten my wallet and had parked illegally. And those wonderful women howled with laughter and told me everything would be okay. They showed me that no matter how hard my journey may be, I would never be alone. I had met my tribe. They showed me how to laugh again, that it was okay to laugh, and gave me the belief that somehow, that although the journey ahead was terrifying, my family and I would survive, and that it was possible to live a life full of joy. I also learned so much about genetic epilepsy from the incredible experts who attend every year and give their precious time to educate and support us. I am so grateful to them in giving me the understanding and the hope that I so desperately needed. My name is Jennie Nowers. I am originally from Scotland and I studied medicine in Edinburgh. In 2007, I moved to South Africa, and it was there that I met my husband, Steve. I now live and work in Brisbane with my husband and 3 sons. I am a doctor. I like to fix things. I have treated children who are very unwell in Africa with HIV and tuberculosis. I look after families, just like yours, and I love my job. But I'm not attending GETA as a doctor, nor as an expert in my field. I am attending as something much more important, no offence, Ingrid, as a mum, as a parent, because with all of my medical training, there are some things that I can't fix. And there are some things that all the training in the world can't teach. And the emotional roller coaster of a journey that is parenting a child with complex special needs and epilepsy is one of them. So I wanted to say to each and every one of you, I see you. Well, I don't, as this is pre-recorded and beamed out virtually, but you know what I mean. Bloody coronavirus ruined my best line. In December 2018, we welcomed our 3rd child into this world. His name is Lucas. His brothers were over the moon. We were all so excited. Everything seemed perfect. Until the next day, the day everything went wrong, the day our lives changed forever, the day the bottom fell out of my world. Lucas had a seizure. The doctors weren't sure it was at first, but I knew, and I had never wanted to be more wrong about something in my life. Then he had another, and another. I have been a doctor for 15 years. I have seen seizures. They are pretty awful to witness, but I have never seen a newborn baby seized until I watched my own son, and I have never felt so terrified and so helpless. When he was 3 days old, Lucas was transferred to the pediatric ICU. As you all know, there is no scarier place in this world, even as a doctor. In fact, I think it made it worse. I didn't know if he would live or if he would die. Lucas had a battery of tests, an MRI, 2 spinal taps, lots of blood tests. Nothing was wrong. Thanks to a research study in Melbourne looking into rapid genome testing, we got a diagnosis when Lucas was 9 days old. Lucas had KCNQ2 encephalopathy, a devastating genetic epilepsy, a diagnosis that shattered the hopes and dreams we had for our son. Not only would he have seizures, but he would also find it harder to do things as simple as hold up his head or sit. He may never walk. He may never talk. He may never learn to read or write. He is unlikely to ever live an independent life. When we finally got our son home after 6 long weeks in hospital, including his first Christmas, I hit the ground running. There may be no cure for this condition, but we would help him in every way we could. Physiotherapy, hydrotherapy, occupational therapy, music therapy, speech therapy, any therapy I could think of. We fought to get him the specialized equipment he needed to live his best life. Most importantly, I came to GETA. I connected with other parents fighting this battle and joined this amazing community that you are so grateful for but never wanted to be a part of. I drew strength from the positivity and resolve of other incredible families advocating for their children. We are proud of how far our beautiful boy has come, but it has been tough, emotionally, financially, and we have a long road ahead. I like to think I can handle most things life can throw at me. I have emigrated twice. I'm a doctor. I have 3 children. I help people. I support them through their hardest times. But the truth is, this has been the hardest journey I have ever faced. There is a grief process that we all endure, and I'm not sure it ever really goes away, You grieve for the child you thought you would have, the life you thought you would live. I'm sure we all have degrees of PTSD from our times spent in ICU watching our children battle epilepsy. And for many of us, facing an uncertain future with many unknowns is very confronting. But along with the pain, there is incredible joy. I still cry when I compare Lucas to his peers, walking, running, talking, and feeding themselves. But when he does achieve something, for instance, he recently rolled over, I smiled for weeks. That joy and that pride is immeasurable. I almost feel sorry for the parents of typical children that they will never experience it. And when Lucas smiles, he literally lights up a room. He brings out the very best in everyone he meets, and he brings them joy just by looking at them with his beautiful big blue eyes. That is his superpower. We also become the most fierce advocates for our children. I know every one of us will have fought the battle with NDIS to make sure our precious kiddos get the help and support they need. Personally, I think I'm a better doctor because of it. I love looking after the families of complex children and helping them navigate the medical system, the NDIS, Centrelink, and their own thoughts and feelings. Because I think I get it in a way I never did before. And I believe that peer support is so important, and GETA plays a vital role in bringing families like ours together, and I know the hard work that goes into that every year. So, I wanted to say thank you to all those who are involved in putting this conference together. They go the extra mile, even driving kilometres out of their way to take me home when the Uber driver couldn't understand my hybrid Scottish-South African accent, or educating me about Adelaide and its abundance of running water and bitumen. I can't wait for the coronavirus and all the added stresses it brings to our families to pass and to have the chance to see you all again. Thank you.
Thank you very much, Jennie. We appreciate you sharing your journey and that of Lucas and your whole family. And it's one I think everyone who's attending today can relate to in so many ways. I'm going to go on now to our next speaker, who's Professor Lynette Sadleir. Before I do that, I want to mention one thing, which is that in a few moments after Professor Sadleir speaks, I'll update you on the results of our SurveyMonkey kind of expression of everything that you tell us. And it's pretty interesting results. So far, about 100 of you have shared information with us. And I think there are far more of you who are participating. So if you do have the time to fill that in, I'll add your data for when we get an opportunity to talk about that in just a moment. But now I want to go to Professor Sadleir. And I've been looking forward to this. Professor Sadleir is joining us from just across the way in New Zealand, across the Bass Strait. Professor Lynette Sadleir joined the Department of Pediatrics and Child Health at the University of Otago, Wellington in 1999, following an epilepsy fellowship at British Columbia's Children's Hospital. She's the director of epilepsy research at the University of Otago and Wellington, and her main focus of research as a physician scientist is identifying new and defining emerging epilepsy syndromes and finding their genetic causes. So that's part of who Professor Sadleir is. But like all of us, she does many things. She's originally from Canada, and I have it on good authority, Professor Sadleir, that in addition to being an outstanding physician scientist, you are something of a swimmer. In fact, a synchronized swimmer with your sister in the Olympics in Los Angeles. I am so impressed on every level with your athletic prowess as a Canadian and now a Kiwi, and also with your scientific heft. So congratulations to you and your sister for that, and for coaching the Commonwealth Games, and for being a star scientist in New Zealand whose research is important around the world. We're so delighted that you can join us, and I'll turn it over now to you, Lynette.
Thank you, Sarah, for that lovely introduction, although I'm incredibly embarrassed, and I'm sure I have Ingrid to thank for that. But moving on, phenotype. What is it and why does it matter? These are the two questions that I'm going to answer, hopefully, in the next 20 or so minutes. And we're going to start off by talking about what is phenotype, what does it mean? But before I can go into that in any detail, I think it's actually really important that people understand what genotype is first. So genotype is the genetic makeup of a cell. And as you know, we're all made up of trillions of cells. So a genotype is the genetic makeup of us as individuals. And for those of you who may not be as genetically onto it as Ingrid and Sam, and forgotten a lot of your genetic teaching, I'm going to give a little basic background here. So we have trillions of cells in our bodies, and the majority of those cells have our genetic code, which is also called the genome. And that genome is bundled up into packages, 23 chromosomes. And each chromosome is made up of long strands of DNA. And what DNA actually is pairs, base pairs, as Sam mentioned, of nucleic acids. And there are 4 nucleic acids that make up DNA. And they are depicted by these letters, T, C, G, and A. And it is the sequence of these letters throughout the DNA which is our genetic makeup or the genetic code. A series of these codes is what we call a gene. And what do genes do? Why do we have them? So within our cells, we have factories that use the code within this gene to make proteins. And there are lots of different genes, 20,000 of them, as Sam's told us, and they code for different proteins. And proteins are really important. They are molecules which are essential. They basically form the function, function and structure of our cells. And groups of cells form together to make organs. And of course, we are just a big group of organs and cells. So the genotype is this part of this slide. It's the genetic code that makes us who we are. And this is a slide showing lots and lots of that genetic code. So this is a sequence of base pairs. So this is the genetic sequence. And on this page, there's about 1,200 of these letters. Which looks like a lot, but as Sam mentioned previously, and I really need to, it's really important for you to understand, is that your DNA sequence is billions of these, billions of them. And what is absolutely remarkable is that no 2 people are the same. And the differences between our codes, the things that are different between you and I, some of those differences make no difference at all. So it might be that you could change this A to a C and it would be exactly the same. It would make no impact at all on the person. Sometimes those changes some of those changes are what are important in making us different from each other, so giving us all the characteristics. And sometimes a change causes a disease, or what we're interested in here is epilepsy. And remember that we all start off as a single cell, and then we grow and we grow and we grow and we grow until we get to be an adult with 40 trillion cells. And the genetic material in that first cell has to be copied over and over and over and over and over. And fortunately, we have some amazing sophisticated techniques in our body to make sure that that happens exactly the same every time. But unfortunately, it doesn't always go perfectly, and sometimes mistakes can be made when our genetic material is copied, so that the copy isn't like the original, and actually it's abnormal now, or it's changed. Now, sometimes those changes can not do anything to us, so they're what we call benign, or they don't have any effect. And sometimes those changes are what we call mutations, or the more up-to-date term these days is pathogenic variants. And so when you have a change, and if it's a pathogenic variant, that the code is changed, so that means that the protein doesn't get formed properly. And then sometimes when those proteins aren't formed properly, that leads to disease or epilepsy in our case. But we're here to talk about phenotype. So the phenotype is the combination of our genetic code, so the genes, and the interaction with the environment, and that makes the phenotype. The phenotype is basically just observable characteristics. And there are lots of different types of characteristics. So they may be physical, they may be developmental and change over time, It might be looking very deeply into a cell, so it could be biochemical, or how those cells interact with each other, so physiological, or a phenotype can be a description of how we interact in our behaviors. When you're describing a phenotype, you can look at it at a very high level, so take a big picture view, or you can delve deeply to look down to minuscule detail and to really describe a phenotype intimately. So I'm going to now change tack off people, and we're going to look at an analogy of planets. So here we've got a picture of space, and this is our universe with our planets. And if we were to describe these planets, we're looking at them and we would say, okay, well, they're all round, but they are slightly different sizes and they've got different colors. And so we could describe these at a sort of big picture level and be able to differentiate them from each other. However, there's a lot more to them than just being round, and we can look in much more detail at one of our favorite planets, Earth, and we zoom in and we can see that there are continents and oceans, and the closer we look, we can see countries and mountains and plants, and as we get closer in and we're more detailed, we can see that there are cities, and those cities have streets, and the streets and parks, and there's houses and cars, and all of these features are important when we're describing our planet Earth. But here's Earth today, but Earth hasn't always been like this. So Earth has changed. So 5 billion years ago, it looked like this, and over time, it has changed in terms of where the continents are and the temperature, and there has been a significant amount of difference over time, and to get to where we are today. But of course, we're not actually talking about, what we want to look at is people, and what we are specifically talking about is the phenotype, or epilepsy phenotype of these people. So we're wanting to look at them in detail. Now, when we talk, we're going to move from an analogy of planets, and we're going to move on to fruit now. So when people use the term epilepsy, they talk about it as being a disease or a disorder. But using the term epilepsy like that is like looking at this picture and saying there is fruit. And indeed, there is fruit on this page, but it doesn't take very much observation to see that there are actually a lot of different types of fruit, and that they can be divided up and into specific fruits, apples, bananas, and raspberries, and that each of those are very distinct from each other and have specific characteristics. So it's the same with epilepsy. Epilepsy is a group of disorders which we can characterize. And just like we look at this orange, and there are things about it that we can tell people to describe it, epilepsy syndromes are the same. So epilepsy is a group of disorder, and we define these disorders or syndromes or phenotypes by the age of onset that the individual presents, the different types of seizures and how they change, the development of the individual, what we find on an examination, other comorbidities that we were talking about previously, as well as what we can see on their MRI and their EEG. And it's all of these characteristics of the individual and how they've changed over time, which are the epilepsy phenotype. So that's the phenotype. Why does it matter? Well, there are a lot of reasons why phenotype, particularly epilepsy phenotype, matters, and I'm going to talk about 3 of them. So the first one is the importance of the phenotype to epilepsy gene discovery. So if we were before the first epilepsy, or even now, if you're trying to find an epilepsy gene, and if you looked in a group of people and with epilepsy, but that you didn't know what kind of epilepsy they had, so that's these gray individuals all have epilepsy, but we can't tell what kind of epilepsy they have, and you were to sequence their DNA and have a look at their DNA, you wouldn't be very successful at finding what is the underlying genetic causes. And that's because you're comparing apples with pears and peaches with plums. What we need to do, and what's been successful for genetic discovery, is to phenotype those individuals into their specific epilepsy syndromes. And once you've done that, you can group people together who have similar types of epilepsy or epilepsy phenotypes, and then look at those to try to find their epilepsy genes. And that has been essential in terms of us moving forward with epilepsy gene discovery. And as Sam mentioned in his talk, historically this has been done with families and phenotyping individuals with those families. And here indeed is one of the earlier families. So this is one of Sam and Ingrid's families that they described over 20 years ago. So they, I think this was another one that was part of Ingrid's thesis. So Ingrid and Sam interviewed and looked at the medical notes and did meticulous phenotyping of all of these individuals. And they recognized that this was a new type of familial epilepsy syndrome, which they labeled genetic epilepsy with febrile seizures plus. And Sam actually mentioned this previously. And so individuals with in these families have quite a mild, most of them have a mild type of epilepsy, where individuals had seizures with fever between the ages of 6 months and 6 years, and sometimes they would have seizures with fever after that, or they would have generalized tonic-clonic seizures without fever over that period. But they were normal individuals, and they did almost all of them did really had a good prognosis. And so it was looking at families like this, which, and indeed I think it was this family that found that SCN1B was found, but it was in families with genetic epilepsy with febrile seizures plus, where they found some of the first epilepsy genes in the sodium channel. And this is a picture of the sodium channel. And this is the inside of the cell, this is the outside of the cell, and you can see the sodium goes through the middle of this channel, which is made up of proteins, and each of these proteins is coded by a specific gene. And the first gene, as Sam said, in these families was SCN1B, but shortly after that, SCN1A was discovered in families with genetic epilepsy with febrile seizures plus. SCN1A codes the alpha portion of this sodium channel. And so to make these genetic discoveries, it was really important for Ingrid and Sam to identify the phenotype very clearly, and as that was what enabled these to be discovered. So that's the first reason why epilepsy phenotyping is really important. It's because it helps find genes. But the second reason is once you've found the gene, and Sam also alluded to this, we're often in a situation where we have to decide whether the change in, so you have an individual who has epilepsy and they have a genetic test and they have an abnormality in that gene, and we have to decide whether or not that actually is the cause of their epilepsy. Because remember, when I show you this slide here, that we all have billions of different base pairs and that all of us are different. And so we need to figure out when we do have a child with epilepsy and we do an epilepsy genetic test and we get a change, we don't know whether that is the change that's going to cause, that has caused the child's epilepsy, or that's just one of these changes as it happens and doesn't have any impact at all. And so phenotyping is really important. Once we've found a gene, we need to identify all of the phenotypes associated with that gene so that we can tell whether or not changes in our genetic results actually are important for the individual. So it would be really nice if we had this situation where you had an individual with a phenotype, you took their blood, you did the sequencing, and you found a mutation in a gene, and that's it. Boom, got the answer, let's all move on. But that would only be the case if a single epilepsy syndrome equaled a single genetic disease. And as Sam's mentioned earlier, unfortunately, life is not that simple. And what we're often left with is we have an individual with a particular phenotype, we do DNA sequencing, we find an abnormality in a gene, and we don't know whether that is important or disease causing. And the reason it is all a bit of a muddle and all complicated is because we have this complex situation where one gene can actually, variations in one gene can actually cause many epilepsy syndromes. And so if we look at SCN1A, so I first of all mentioned that the first epilepsy syndrome that SCN1A was identified as causing was febrile seizures plus, but subsequent to that, we know that SCN1A abnormalities also cause developmental and epileptic encephalopathies, and the DEEs, which Sam mentioned, and these are a severe group of epilepsy that usually presents in children. And in these group of epilepsy syndrome, the frequent seizures and the lots of epileptic activity happening in the brain between the seizures means that the brain can't develop properly and end up with a majority of them having a poor prognosis. And indeed, SCN1A causes more than one type of developmental and epileptic encephalopathy. It causes Dravet, which I hear from Sara that many of you out there have children with Dravet syndrome, so you will know this disorder well. But the children with Dravet syndrome present at 6 months of age with seizures that are long, often one-sided with a fever. And about 80% or more of children with Dravet syndrome, it is caused by mutations in the gene SCM1A. Another epileptic encephalopathy that SCM1A causes is epilepsy of infancy with migrating focal seizures, which is different from Dravet syndrome in that the children present earlier and they have different types of seizures. And this is what, so not only, however, do we have a situation where one gene causes many different types of epilepsy phenotypes, but we're also, it's confusing and complex because some epilepsy syndromes have lots of different genes that cause them. So in the situation with epilepsy of infancy with migrating focal seizures, yes, it can be caused by SCM1A, but this is the circle of etiology of this syndrome, and only a small portion of it is caused by SCM1A. The biggest portion is caused by mutations in genes called KCNT1, but you can see all these other letters representing other genes that are also involved. So you've got this complicated situation where one gene causes lots of syndromes, and some syndromes can be caused by lots of different genes. And so therefore you have a problem sometimes when you get a genetic result, knowing whether the genetic result is actually important to the individual that it's been done on. And that was the situation in this young man here as clinician. So one of my colleagues looks after this young boy, and he has severe epilepsy, and she did an epilepsy genetic test. And remember, these days when you do a genetic test, it's looking at variation or changes in lots of different genes that have been known to cause severe epilepsy, so over 100 of them. And so the result came back and it showed an abnormality in SCN1A, but he didn't have Dravet syndrome, and he didn't have epilepsy of infancy with migrating focal seizures, and he definitely didn't have febrile seizures plus. So she wasn't really sure what to make of that. Was it just a change in the SCN1A gene which didn't cause disease and it was irrelevant, so it was a red herring, and was there something else that caused this child's epilepsy? She wasn't sure. She was asking what I thought. And it was a few days later after that Ingrid and I were having a conversation, and she's looking after a boy who had a similar type of epilepsy, and she had had the same thing happen. She'd done a gene test and it had an abnormality in SCN1A. And so we got to thinking, well, perhaps there was another type of epilepsy, severe developmental and epileptic encephalopathy phenotype associated with SCN1A. And we were able to put together a group of children from all around the world. And indeed, they did have a quite specific phenotype. They presented with seizures under the age of 3 months. They had frequent seizures, and they ended up with poor prognosis, with being unable to walk, unable to talk, and most of them had G-tubes. And they had this, so they weren't able to feed. And they had this quite specific movement disorder as well. And so these children are just constantly active and moving around. So because of putting these groups of individual children together, that meant we were able to identify a new epilepsy syndrome associated with SCN1A. If I can just move, and that is early infantile SCM1A encephalopathy. And so now when children have this phenotype and they do an epilepsy gene test and they have a variant in SCM1A, the clinicians will be able to say, yes, this is likely to be the cause of this child's epilepsy. So it's not just important to have phenotyping in terms of finding the genes, but once you have the gene, it's really important to keep looking and finding other phenotypes that this a specific gene causes, so that when people do the tests, they can make sense of that result. And then the final reason that I'm going to talk about why phenotyping matters is really the most important reason, because as Sam says, it is if you have severe epilepsy and you get a genetic gene test and you get a variation in a specific gene, and you've decided, and your clinicians have decided that that is probably the most likely cause of your epilepsy, then you have a genetic diagnosis, and that's wonderful because it gives you an answer and because it means you don't have to keep looking for a cause. But at the end of the day, it is just a bunch of letters and a number. And what is much more important is, what does that mean? So what's really what people want to know when they get a genetic diagnosis is, so what's going to happen to my child, and what is the best way to manage and to make sure that my child has the best quality of life that they can have. And so once you find a genetic epilepsy, you need to keep doing research on larger and larger groups of them, so you can define the phenotype in more and more detail. A little bit like what we did in the planet Earth when we're going zooming down to learn about the houses and the cars. So I'm going to give you an example of how we do that, or with PCDH19, or girls clustering epilepsy. And so that was identified. Again, this is another one of Ingrid's families in 2010. And this is an unusual epilepsy or inheritance in that only girls, so although the girls are in pink, that pink actually represents that they have the disorder. And this is a type of epilepsy which is just found in girls, or mainly in girls. And these girls present with seizures around in the first year of life, and they have clusters of seizures. And so that means is they have multiple 10, 20 seizures over a 4 or 5 day period. And the seizures are quite are similar in that they often have the children look frightened or might scream. And it can be very difficult to stop the clusters, so to abort them. And it can also be difficult to prevent the clusters coming in these girls. And over time, although the girls start off with normal development, about 2/3 of them end up with some sort of intellectual disability. And so this was discovered in 2010, and so it's now 9 years later. Lots more individuals have been diagnosed with this genetic epilepsy, and so we have bigger groups of people to find more and more out, and to find look deeper in the phenotype. And so more recently, we were able to find that one of the a feature of PCH19 girls clustering epilepsy is that, unfortunately, about 20% of them in late childhood or adolescence and early adulthood, they develop psychosis, and about 15% can actually have schizophrenia. And so that's an important thing to know, so that people can plan, they can look for that disorder and can make sure they get treatment as early as possible. And another thing that we've been published recently, just this year actually, is we've been able to look at girls with PCH19 girls clustering epilepsy, and have identified that levetiracetam in some of them can be quite miraculous. So both Ingrid and I identified that some of our girls, when we put them onto levetiracetam, their epilepsy just seemed to stop. Now that doesn't happen with all of them. And so we looked at a larger group of girls, and were involved in a survey with Christy Kulk and Joseph Getz, which many of you who may be listening might have been involved in. And we were able to show that, yes, levetiracetam does indeed seem to be good for girls with girls clustering epilepsy. So this is an example of deeper phenotyping, and it really is helpful for the families. So that's the end of my talk. And so in conclusion, I hope I've answered these two questions. So what is phenotype? It are the characteristics which are determined by the genotype and the environment. And why does it matter? Well, it's essential so that we can identify genes, so that once a gene is identified, we can determine what phenotypes are relevant to that gene. And ultimately, and most importantly, that we can give good, accurate information about what happens to individuals with the those genetic disorders, and how we can manage them best. So I'd just like to thank you, Ageta, for putting on this symposium, for inviting me to speak, and I'm happy to answer any questions.
Thank you so much, Lynette. I think we do have some questions, and we really appreciate your presentation and your willingness to answer some of those. Speaking to the international nature of this conference, one of our questions is from Nicholas in Germany. And the question is, which specialty, or who looks after children with genetic epilepsy variants without seizures? So for example, SCN2A, loss of function. So what happens when you don't have the seizures, but you do have the phenotype?
So who looks after them? So I guess Ingrid and Sam and I are what we call epileptologists, but we're neurologists first off, and then we've sort of specialized in epilepsy. So our research and our clinical load, or clinical patients that we see, individuals have epilepsy, but neurologists, and in some places developmental pediatricians, and it depends on what the actual phenotype is. So if it's a phenotype that's a psychiatric or mental health, then psychiatrists. But different clinicians have different expertise, and but so if you have a genetic disorder and you don't have the epilepsy phenotype, then an epileptologist is probably not the right person to look after you. But there are neurologists and psychiatrists and behavioral developmental pediatricians who also have expertise, significant expertise in genetic diseases, that are able to manage individuals who don't have seizures.
Well, and it's interesting as well, Lynette, because when you talk about phenotype, I think you laid out really nicely, or with great detail, how complex that can be and what those needs can be. But the truth of the matter is that some of these phenotypes are difficult. They're complex and they're long lasting. So as we learn more about genetic epilepsy and we understand these phenotypes more, it can be a lot for parents, particularly in those early days when a newborn or a very young child has been diagnosed. What do you think needs to be done in terms of supplementing that understanding of both genotype and phenotype with the kind of array of support that a family can and should get as they begin that journey?
Well, I think that that's a really good question. And I think that once you have a child who has some sort of a disorder, epilepsy in this particular situation, and then you get a genetic diagnosis, as I mentioned in the talk, understanding what that phenotype is means that so basically what that means is just you're giving some information about what's likely or possibly going to happen to your child. What's the outlook going to be? And of course, we can't say absolutely, but we can get a bit of a rough idea that other children with that kind of variance, this is what happens. And so because you get that information and your clinicians get that information, it gives people a bit of a forewarning as to what might happen and what help you might need and what therapies might be best for you, not just drug therapies, but other therapies as physical therapies and also other areas of support, be that medical or family support groups like yourself or financial. So it really allows people to plan better and to be better prepared. And I know when Jennie gave that fantastic PowerPoint and how devastating it was, and many of you families out there will know exactly how that is. Sometimes just some information and knowledge about, potentially what happened is helpful in that time to be able to think about the future.
Thank you so much for that, Lynette. Maybe just a couple of more questions and then we'll let you go. This is from the UK. Does one gene mutation, you touched on this, does one gene mutation cause different presentations? It's clear that it does, for example, SCN1A, but I found this interesting, even in the same family.
Yes.
That was interesting.
Okay, so there's two aspects. So you that's right. So you can have one gene with lots of different variations in different places, which can cause different phenotypes. But what I think they're alluding to is you have one gene and you have one mutation in the same place. And yet you have slightly different phenotypes with that. And so that's because we are more than that one. Remember, we've got billions of code, and the other billions of codes are different between people. So the impact of one change can be different based on what your overall genetic background is. So if you have, and a bit like what Sam was talking about when he was mentioning about the height, the polygenic risk score, so that person had lots of them that made them really, really tall. Well, when we're talking about a genetic mutation or just one change, it's not on its own, it's on the background of all the other genes and everything else that's making us who we are. So it's that impacts on what we actually see and why people aren't all exactly the same, because they're not all. Nobody's exactly the same with all those billions of codes.
Exactly. It does get complicated. And I think that's why it's great to tease it out as you're doing just now. And as we leave, and this is kind of related to that, this will be our last question. What about mosaic mutations and how can they affect phenotype?
Okay. Oh, what a fantastic question. Okay, so mosaic, for those who don't know, is when you have the change in your DNA occurs after the cell and the egg get together. So every cell in your body doesn't have the same genetic code. Some cells have a change and other cells don't. And so if you have, and so that's why, so if you have a mosaic change, sometimes the phenotype is not as severe because every cell doesn't have that change. So some of the cells actually are working better because their genetic makeup is normal. And so sometimes we have mosaic phenotypes which are just milder than the phenotype if you had the genetic mutation in all of the cells. I think that's probably the best answer for that question.
All right. Thank you very much. So Lynette Sadleir, we really appreciate it. Professor Sadleir, there's such a great expression and understanding of phenotypes, which are very important in our community, because it's not just about the genetics, it's not just about the letters and the numbers, but it's definitely about the phenotype, that drilling down into the specific things that all of us can expect in terms of how our children are going to grow and develop. So thank you so much for joining us from the University of Otago in Wellington, New Zealand.
Thank you.
And as we get ready to give ourselves a little bit of a break for what is morning tea here in Australia, and maybe a middle of the night power bar for some of you, or possibly it's dinner time, or maybe you're even having a glass of wine. Well, we welcome you, whatever you're doing and wherever you are. But before we go on to that little break, which will be in just a few minutes, we want to talk about two other things. I want to give you a little bit of the results from our SurveyMonkey. And then before I do that, I want to introduce you to one of our other family members who's got some prepared remarks for us. We talk a lot about what the impact of having a genetic epilepsy is on parents and also on siblings. But sometimes we forget about grandparents. So what we'd like to do now is hear from Bob Pierce. And he's the dad of Chris Pierce, who's one of our family members here. And he's going to talk about being an SCN2A grandparent.
Hi, my name is Bob Pierce. I am grandfather to Will Pierce and my 7 other grandchildren, but known to all of them as Pop. When I was asked to prepare this talk, I thought it would be a piece of cake. How wrong I was. It has unleashed a flood of memories, both heartwarming and heart-wrenching. My story started back in 2001 when my daughter was due to give birth to twins, Ella and Will, and my wife and I were asked to be present at their birth. From the start, we knew something was wrong with Will. Not quite right. And what eventuated was, after many consultations with many doctors, Will was finally admitted to intensive care, where he was hospitalised for approximately 6 months. It was during this time in hospital that he was diagnosed with severe epilepsy. On being released from hospital, both Jude and myself took a role of caring for both Ella and Will. And as I worked, Judy was the primary carer, with her travelling to look after them. Over the years, we struggled with Will not being the same as other children, which meant many trips to the doctors, specialist, and stays in hospital. This was until aged 14, when we were finally given a diagnosis to Will's rare condition, which was SCN2A. Whilst this was a relief to finally have a diagnosis to identify with, it brought new challenges. Constant changes in medications, learning to use EpiPens were just some of the challenges we faced. We assisted in picking up from school and looking after the twins when Mum and Dad were otherwise occupied. And these were just some of the tasks we undertook to assist in their upbringing. It has also been a great pleasure to be part of Will's many loves over the years. These include planes, trains, hamburgers, Queen, the band of course, and of course his great love of Highland cattle. And it's certainly been a great pleasure for me to take Will to many country shows where Highland cattle were being shown. His infectious personality has meant that he has been embraced by the Victorian Highland Cattle Breeders Association, to the extent that one of the breeders has taken him under her wing and taught him how to wash, groom and prepare cattle for showing. This has resulted in many trophies being won by Will, and as I said, it's been a pleasure to be able to travel with Will to these shows and be part of his life in that. So as you can see, my journey with Will has not only been as his pop, but one of sharing 18 years of growing with him. And all I can say has been an absolute delight. Thank you for letting me share my story.
Bob, thank you for sharing your story. It's so wonderful. I think all of us here at GETA know Will, and we've had the opportunity to hear from him at past conferences, and his love of Highland cattle and his love of life is really something, as is the adoration of his entire family. So thank you very much for providing the perspective of a grandparent, because genetic epilepsy affects us far more broadly than people sometimes imagine. I think sometimes we say that we're a bit of a club. We didn't ask to join, but we get it. And that extends far and wide throughout the families. And speaking of the families, we're going to take a morning tea break. But before we do, allow me to tell you a little bit about you. We have the latest results from SurveyMonkey. And as you know, there are a lot of you who are joining us from here in Australia. We're in the correct time zone, plus or minus a couple of hours. And you really are from across the US. Also, as we mentioned, the UK, New Zealand, North America. We talked about some of the other countries around the world. But I thought you might want to know what some of the answers are that you're giving for why you're taking your time to participate with us today. And there's one of the greatest responses is want to hear about the latest treatments and hear about the latest progress towards a cure for genetic epilepsy. And of course, some of those cures will happen probably one at a time, and probably a bit of a three musketeers approach, all for one and one for all, although there are more than three musketeers by this stage. There, as we have learned already this morning, are well more than 100 different genetic epilepsies. But I think every time we see progress or a treatment, even if it's not for our specific variant, it really does give us hope. And speaking of which, hope is another reason that you're joining us, and another is to connect with other genetic epilepsy families. So we encourage you, stay in touch with us. Let's talk about what the genetic epilepsies are that you or a loved one have. As we mentioned before, Dravet or SCN1A is really a large share of that. But then, as we just heard from Lynette, that can really be broken down, SCN1A proving to be far more complex than anyone had originally thought. Also CDKL5, SYNGAP, KCNA2, KCNQ2, SCN8A, SCN2A, LGS. These are just some of the ones that we have here today. Here's another statistic that I found very, very interesting and indeed quite moving. We have families who are joining us who are with children of all ages. That starts at newborns. We have a large percentage there. Probably our biggest percentage is those of you who have children who are in the age of 6 to 10. But by no means is it you alone who are participating today. We have everyone represented in terms of families, from those of brand new babies all the way through to older than 25 years old. And so a real hats off to you who have older children, because you're staying with it. You really prove the depth of care and love and response and trying to find out the latest information. So we give you extra credit, and also to you new parents who are starting so early. So all along the journey, we're glad you're with us. So those are just some of the pieces of information that we're able to provide from what you're telling us. So the more you share, the more we can reflect that back to you. We're going to take a slightly shorter morning tea break, or whatever that break is, wherever you are. So we'll take about 5 or 10 minutes, and then we'll be back, and we will continue when we return with Professor Ingrid Scheffer. She's going to talk about new treatments for genetic epilepsy. So you're not going to want to miss that. The good, the bad, and the unproven. So that's what we're going to have when we come back in about 10 minutes. See you then. Welcome back, everyone. We hope that you had an opportunity to get a beverage of some sort. I have a nice cup of tea, and in fact, this cup of tea is significant in terms of our next guest, because it's really faded, which just goes to show how long I've been in Australia, which is 12 years now. But if you have really good eyesight and maybe a little bit of imagination, you can see that it says, no, you really can't see that it says, you're gonna have to take my word for it, that it says Today Show. There you go. Look, you can see that. Today Show. This is today. on NBC. Except it isn't. We're right here in Melbourne, Australia, and we're talking about genetic epilepsy. But long ago, in a country far, far away, when I worked for NBC and actually filled in a lot on the Today Show, I was pregnant and I had a really cool daughter, and then I got pregnant again and had another really cool daughter, except that with our younger daughter, we learned that she had something wrong. And when we later moved to Australia, I realized that it was very important to find whoever the best expert was to try to find out what was wrong with our daughter. So I treated looking for a cure and looking for a treatment like a news assignment, as if I were following the leads that way. And I've always believed that it's important to get the best. And so I asked our doctors in New York City who was the best in Melbourne, Australia. And that's called hitting the jackpot, because it turned out that the expert to whom they recommended us was not just a great expert in Melbourne, but also an international expert. You've already met the other half of her scientific partner, Sam Berkovic, because Ingrid Scheffer, along with Sam Berkovic, proved that non-inherited epilepsy could be genetic. And of course, they did that with the other experts that we mentioned in Adelaide and Germany. But she is not only an outstanding scientist, she has become a dear friend over the years. She said I have to talk quickly, but I've known her too long and I'm not going to. So I'll just tell you that she's among her many awards. If she were in the military, she'd have a lot of brass and ribbons, but instead she gets these scientific honors which don't show up. So I'll have to tell you that she was a L'Oreal UNESCO Women in Science laureate, that she's an officer of the Order of Australia, and won, along with Sam, the Prime Minister's Prize for Science. Those are just a few. Take it from me, she is as kind a doctor and an optimistic doctor she is an astute, shrewd scientist. Among the recent things that Ingrid has done, she led the first major reclassification of the epilepsies in 30 years, and she also published a big study that shows that medical cannabis is effective in epilepsy that was published in the New England Journal of Medicine. So it is my pleasure to welcome, with a long introduction because I've known her so long, Professor Ingrid Scheffer, who's going to talk to us about new treatments for genetic epilepsies, the good, the bad, and the unproven. And she's right close to me, so I might have to move away so she doesn't hit me. Ingrid, I'll let you take it away.
Well, thank you, Sarah. Thank you for that lovely introduction. And I'm very pleased to say I'm 1.5 meters away from you right now. So I thought I might touch today on new treatments for genetic epilepsies, the good, the bad, and the unproven. But before I do so, I just wanted to really say how fantastic and how exceptional the families are that really are behind GETA. We have the 3 main families here today, but there are other important families that contribute in New South Wales and in Adelaide and other parts of Australia. And I think that you all need to be congratulated on your amazing vision in bringing all the families together in Australia. We're a small community, and that means we can't get 1,000 patients with one genetic disease, but we can get a lot of families who are really facing very similar problems. So congratulations on your vision, and I'm hoping that next year we won't have to be 1.5 meters apart and we can all be together. And also the conferences for the last 4 years are all online. So if you want to hear more, there's masses you can listen to. So I wanted to touch on new treatments for genetic epilepsies, and really it's still very early days in this space and we have a lot to understand. I'm going to tell you about the good, the bad, and by that I mean those that people are saying are good, but we really don't have scientific evidence. And finally, and importantly, the unproven, and that's the watch this space part. Now, there's already been a lot of excellent talks talking about the severe epilepsies, the developmental and epileptic encephalopathies. So I just have one slide to make sure that we're all on the same page. These children have frequent epileptic activity on their EEG. They usually have frequent seizures, often many types of seizures. They have developmental delay often before the seizures start, but sometimes they don't. For example, in Dravet syndrome, they don't. And then they show developmental slowing and often regression, but not always regression. So that's the group of disorders we're talking about, the developmental and epileptic encephalopathies. encephalopathies, and there's well over 100 now different genes for these diseases, and these are really each separate diseases. So I'm going to focus my talk on Dravet syndrome because really it's the poster child for this group of diseases and we have the most information about it. But a lot of what I say will be important to think about how we can use it, this information, this approach to your child's disease. So, for example, we think about these diseases as complex puzzles, and in Dravet syndrome, the child presents at around 6 months of age. They're usually well and developmentally normal, and we know that's not the case for many of the other ones, but for Dravet that's typical. They present with a long seizure with fever, which may be jerking down one side or perhaps all over. Often it can be 30 minutes, sometimes it's short. And they go on in the first 6 months of life often to have frequent convulsive seizures, but they may only have one or two. They then go on between the age of 1 and 5 years to develop multiple other types of seizures, focal seizures, myoclonic jerks, absence seizures. And here you see a little boy from Tasmania who at the age of 21 months was having the flashing lights during his EEG, and you'll see that it triggers a cluster of jerks, and at the end of it he lets out a loud cry because he really doesn't like how this makes him feel. Children with Dravet syndrome are normal in the first year of life, but then their development slows. They may go backwards with various seizures, and they usually end up with intellectual disability. Examination in the young child might be normal, is usually normal. They might be a little bit unsteady when they walk, but then as they grow older, they develop this pattern of a crouch gait that we studied with Jill Rodder some years ago to look at how this gait works. And now we see that in quite a few other disorders. I see it in patients with GLUT1 deficiency. I see it in patients with Angelman syndrome, but also I suspect other different genetic diseases will have this unusual gait. And then they walk just a little bit late at about 18 months, and they might develop some more unsteadiness as they get older. The EEG is surprisingly normal in the first couple of years while they're having lots of seizures, but then becomes very abnormal. And finally, and you've already seen this picture from Lynette, the sodium channel gene comes to the fore, and this is where we know that more than 80%, probably more than 90% of children with Dravet syndrome have a mutation that involves this sodium channel. And just to go back to the channel picture, here you see the channel sitting in the cell membrane, and I'm pointing to where the sodium ions go through the channel, and here is the membrane of the cell. And this little picture fits right here, and you can see how it could be very important in the clockwork of one neuron or nerve cell talking to another one. So all of these features together, this whole constellation makes up a syndrome known as Dravet syndrome. And there are now many, many patients around the world known with Dravet syndrome. And I'm just wanted to introduce that before I go into the different drug treatments we're talking about for Dravet syndrome, and some of these may apply more widely. So this is what we want. We want a magic bullet. So if any of you have got that up your sleeve, please send it directly, because that's exactly what we want for these children. We want a magic bullet that will stop seizures, that will help development, that will reduce mortality rates, that will reduce gut problems, behaviour problems, autism, a whole host of issues that are normal in most of the children with the developmental and epileptic encephalopathies. And so I'm going to tell you about the good, where we have proven that there may be some real benefit from medication. The first one for Dravet syndrome, called phenfluramine, is almost a magic bullet, which is fantastic. We don't know the long-term effects yet, but that's what we want. The second one, and the most of my talk will focus on cannabis or cannabidiol specifically, is not a magic bullet, and it looks about as good as some of our, or many of our other drugs for Dravet syndrome, and I suspect in many of the other developmental and epileptic encephalopathies. So we want a magic bullet, but we haven't really got many magic bullets. But that is what I hope Steve Petrou, who speaks after me, will talk about, which is gene therapy, where the real promise lies. So let's turn to the good. And the good we can only talk about when we have scientific evidence, and we draw on that from double-blind, randomised controlled trials. Now, what that means is not like the picture shows, two blind scientists talking to each other. It means that we do a trial where I give a family a medication. I don't know if I'm giving that family drug or placebo being like a sugar pill, and you don't know, the families don't know, the doctors don't know. It's all done in a blinded way by a pharmacist. That's double blind, and that way we can't introduce the bias that we all have in wanting to see a drug make a child better. So it's really important, if you're going to get scientific evidence, that you have placebo versus a drug. Most of the trials that we do in 2020 are short term, and that is only 3 months. And you know, with your child, the life of their seizures and their disease is years. And as we just heard from Will's grandfather, 18 years of trying to find a way to help him medically and help his life be better. So the primary endpoint of most trials is seizure reduction, and they may have secondary endpoints which are looking at what else did it help? Did it help their quality of life? But does it help their autism? Does it help their development? Most trials don't even begin to tackle those crucial questions. And then trials also look at adverse effects. Mild to moderate ones, they're ones that we can live with, you put up with if the drug works. Severe ones may not be tenable with keeping on the drug. You may have to give up the drug trial. And the crucial figure that we use in drug trials in epilepsy is called the responder rate, which is, and this is defined as a 50% reduction in seizures. So you take a baseline period prior to the trial, then your 3 month of trial, and you say, was there at least a 50% reduction in seizures? And that's what I'm going to be talking about in the examples I'm going to share with you. So I mentioned at the beginning, this may be the closest thing I've seen to a magic bullet. And this is a drug called fenfluramine. It's specifically for Dravet syndrome, though there are trials on Lennox-Gastaut now. This is an amphetamine-like drug, and it was brought out in the early 70s as an appetite suppressant. But then they found out that it caused thickening of heart valves and pulmonary side effects, lung side effects, and so it was pulled off the market in 2001. But some very astute doctors in Belgium had noticed that it was good in their patients with Dravet syndrome. So there was a royal decree from the Belgian king permitting the ongoing use, and to the credit of the Belgian paediatric neurologists, Dr. Keulemans and Dr. Legat from Belgium, they have both championed this drug and brought it to pharmaceutical companies who have now run with it. And here are the super exciting data. They just came out at the end of last year. And you can see here, and I'm going to show you quite a few pictures a bit like this. These are 2 different doses of fenfluramine. And we're looking here at the percent difference. So how much better do the children who got drug do compared with those from placebo in terms of mean monthly convulsive seizure reduction? And you can see on the higher dose, 64% of reduction in mean seizures, and in the lower dose, a 33% reduction in seizures. I mentioned responder rate before to you because I think that's really a useful figure, and you can see at the higher dose, there was a 70% of children had a 50% seizure reduction. Remember, most drugs, 30 to 40% is the responder rate we look at. So 70% is fantastic. Does that make a difference to the children's cognition, to how well they are? Well, the parents will tell you they're a bit brighter if they're not having seizures, they're doing better. But are they normal? No, it doesn't bring us back to normal. The other exciting thing about this drug is seizure freedom or near freedom rates. And if you look at the top line here, the higher dose, we have 25% have no seizures or only 1 seizure, which is amazing and really what we're looking for. But does it last? Well, here's the answer. 18 months out, and our data show, yes, it's sustained. So this is super exciting stuff and transformational for patients with Dravet syndrome. But the other side of the coin, does it work? and what side effects does it have? So side effects, yes, it has side effects. And here in this picture, you'll see the bolded ones are the most important. Decreased appetite. Here, in about, on the higher dose, almost 40% of the children. Tiredness, 10%. And in the other trial, another trial with stiripentol, again, 40%. So it's hard because any of you who have Dravet children will know mostly they're not great eaters, they're very active, and so decreased appetite is a worry and something we have to watch very carefully. So what did the families think? Well, the families thought those on the drug, so those in the green bars here, they actually showed a clinically meaningful improvement, and the investigators thought that as well. But I think the families are much more important for this, and here they showed, yes, we think this is making a big difference to our child's quality of life. Very much improved or much improved. So that's all we have at the moment in terms of our almost magic bullet, and I'm hopeful that gene therapy will be another level up from that. But let's now turn to cannabis in childhood epilepsy, and I'm going to talk about the good, the bad, and the unproven. So cannabis is a plant, as I'm sure everyone knows, and a plant contains like 400 different substances with a range of cannabinoids and other compounds in the cannabis plant. In particular, there are two main species that people are interested in. Cannabis sativa, that has a higher THC, which is tetrahydrocannabinol, to CBD ratio. And this is the plant that actually people like because it's psychoactive, it makes them high, and it has stimulatory effects. But it's also the THC that puts you at risk of an earlier onset of psychosis if you may be predisposed to developing schizophrenia in later life. And that's an important point that we shouldn't forget. The cannabis indica plant is more associated with sedative effects. Now, prior to all the hype around cannabis, and I'm sure everyone is more than aware of that, there were in vitro and in vivo seizure models. So that means in the test tube and in the animal models, evidence suggesting that CBD, the cannabidiol, actually has anticonvulsant activity. So this was the first big study of cannabidiol in epilepsy. And there are really only 2 randomized placebo-controlled trials for cannabidiol in Dravet syndrome and also in Lennox-Gastaut syndrome. And it's watch this space for the rest. So I'm very excited to tell you about this one. I was a member of the team that was involved in this study, and it came out in 2017. The company is GW Pharma and their drug is Epidiolex. And this is 98% cannabidiol, and it's from an extraction from plants. So it's natural cannabis. And the important words are at the bottom here, pharmaceutical grade. So, you know, every time you give that child that cannabis, you're giving them exactly the same amount of drug. And here are their findings in the Dravet study. You can see here in convulsive seizures and here in total seizures that there is a significant reduction from placebo. So placebo in green and drug in blue, and 39 to 41%, around 40% of patients showed around a 40% reduction in seizures. And if you look at all seizure types, not just convulsive seizures, the figures are very similar. Turning now to the responder rate, which is the 50% seizure reduction, you can see here, this middle set of bars, 43% of patients were responders. So very similar in Dravet syndrome to drugs such as topiramate and other medications tested for, and stiripentol, other drugs tested for Dravet syndrome. Not better, but certainly as good as. What about the caregivers? What did they think? Well, they liked it too. And you can see here slightly improved, much improved, very much improved. And the blue bars denote the CBD, the cannabidiol, and show it is significantly better than the placebo. So what about CBD, does it have side effects? And the answer is yes, but they're usually quite manageable. So 36% of patients actually were tired. Somnolence meant sleepy tired. One that was a new one in this world is it was that 19% of people have diarrhea, and diarrhea isn't seen very often with the other anti-epileptic drugs we use, so that's a new issue. Again, this appetite suppression, 31% had diarrhea and 28% had decreased appetite. So it's good, but it does have some side effects, just like all the other drugs we use. Just recently, we published a paper in JAMA Neurology, just in March this year, which compared 2 doses of CBD in Dravet children. And interestingly, we found that 10 milligram per kilogram per day, which is half the dose we usually aim for of 20 milligram per kilogram per day, was basically just as effective if you look at these reduction in seizure freedom, and if you look at the in the total seizures and the convulsive seizures. So actually, maybe we can use lower doses, which is important when we think about the expense of these disorders. That's Dravet syndrome. What about CBD in Lennox-Gastaut syndrome? And I'm sure there are many people on the line who have children with Lennox-Gastaut syndrome. Lennox-Gastaut syndrome is a well-recognised syndrome, but it has many different genes that can cause it. And here you see very similar findings, which is encouraging. And it was published in both the Lancet and the New England Journal of Medicine, 2 different papers, same drug, Epidiolex, and you can see here very similar figures with a 44% mean reduction in monthly seizure frequency in the CBD. And here they did the same trial of the 10 versus 20 milligrams per kilogram per day of cannabidiol, and you can see that the higher dose is really not that different from the 10 milligram per kilogram per day. That's the good. Let's think about the bad. What about the bad? Well, I'm not saying that these are all bad, but I do think we need to acknowledge that there is a burgeoning cannabis market in Australia and the world. And there are now many, many companies who are growing their own in Australia, and Australia is keen to get into the pharmaceutical space. But there are issues, and we need to make these companies do the right thing and do the trials to give us scientific evidence that their drug is working. So one of the issues is that apart from the Epidiolex that I've just presented to you, all the others are nutraceutical grade, and that is a bioceutical, which is a pharmaceutical alternative which claims physiological benefits. In the US, nutraceuticals are largely unregulated. They exist in the same category as dietary supplements and food additives by the FDA. And I think it's just important to realise that we're not comparing apples and pears. The other drugs that you're getting from your neurologist or your paediatrician are pharmaceutical grade, not nutraceutical grade. And I did touch on placebo a moment ago, but it's very important to recognise the importance of placebo effect, and you haven't taken that into account if you haven't done the proper trials. A placebo effect is thought to be about 33%. So if I, the doctor, give you, the patient, a drug and say it will work, and that drug is just a sugar tablet, in 1/3 of people it will work because they believe it will work and I believe it will work. And that is not science, that is around psychological factors, which are critical in what we do in looking after our families. Interestingly and intriguingly, the placebo effect is greater in paediatric trials than in adult trials. So with these many forms of cannabis available, the formulations are all nutraceutical grade. They have inconsistency between the formulation, between one bottle and the next bottle, and they may have more THC. Sometimes the other components are known, often they're unknown, and then this leads to this mythical entourage effect. An entourage means what's going along with this? Maybe there are terpenes and flavines and a whole lot of other components. Maybe they are doing good, but we have no proof they're doing good. And yet people want it to say this is making the difference. Well, we need science behind that. So there's definitely anecdotal improvement, and that's fantastic, but I'm just trying to caution that. What about the side effects? And one of the ones that troubles me the most is this issue of THC, tetrahydrocannabinol, being associated with, proven scientifically to be associated with younger onset of psychosis. And what happens if you give your child something with THC when they're 5 years old, and when they're 15 years old, they become psychotic? How do you know that it wasn't that drug? So again, cautionary. The other point to make is it's expensive. At the moment, our governments, for the few children that can access it, are paying something like $30,000 to $50,000 a year for that child to have CBD. It's worth it if it works, it's magic. But if it doesn't work, there are problems. So my challenge to all those cannabis companies out there is please, please do those randomized double-blind placebo-controlled trials so that we can have scientific evidence and be happy and comfortable with prescribing your medicine. So let's just turn to the unproven. And the unproven is, gee, I hope this works. Does it work? I really need to know. And I wanted to tell you about a trial that Lynette, Sadleir and I have just finished with a company called Zenurba. It's a US company, and they make a CBD gel. It is 98% CBD. It's synthetic. It's, I think actually it's 100% CBD. I think my slide's wrong. And what we have done is just the opening ambit, an open label 6-month trial in children with developmental and epileptic encephalopathies. And it was about 48 children from memory. And you can see here the comparison between this 6-month trial, month 3 and month 6, and our responder rate. Now, this is open label. This is not good science, but it's very promising and exciting. 55% of our patients were responders, more than 50% seizure improvement at 6-month place. And this data is being shown this week in the US at the online American Academy of Neurology. So the doctors there are seeing these data there, and they're not published yet, but they're in the public domain. And we also looked at what the families thought in our almost 50 patients, and we used this good day, bad day ratings of the family that Lynette was very keen for us to use. And you can see here the baseline and month 6. And you can see in the purple and in the darker purple, we have a good day and a fantastic day. So the families thought this gel made a difference. Were there side effects? Of course there were. And the main thing was about rubbing this gel onto the arms or the legs. And you can see they could have application site dryness or pain. It's only around 8%. Decreased appetite 4%, but only one of the patients discontinued. So CBD gel, is it proven? No, it's not proven, but now what the next step is to do a randomized double-blind placebo-controlled trial for children and hopefully for adults with developmental and epileptic encephalopathy. And I know that we're thinking about doing that very, very much in the near future. So in conclusion, new therapies for genetic epilepsies, where's the big promise? Well, the big promise is gene therapy, and I will move the floor to Steve Petrou, who will tell you about that in mouse. But there are a few things. The first thing, each gene is a different disease. There is very promising gene therapy in animal models, in mouse, for SCN1A, SCN2A, and the potassium channel gene KCNT1, but also in other diseases. There's huge steps to translate these findings from mouse to man. And in order to be ready, to be poised to see, do these gene therapies work, we need natural history studies. And this is very much what our group has been working towards with child neurologists from around the country. And we are just embarking on a nationwide and hopefully global, but more nationwide, we're going to start small, go bigger, study to do natural history studies of all the severe developmental and epileptic encephalopathies. No matter what gene your child has, we're interested. So if you're interested, please email us on that email that I'm showing you now, and we'll get your name ready for when we're ready to go. We're going to bring in EEG, MRI studies, your information, And why this is important is that natural history data will be the baseline, if you like, the placebo, to compare whether the gene therapy stops the child getting sick and helps the child to lead a normal life, which is our dream. So I'd like to conclude by thanking the fantastic team I work with. You've already seen Sam before in person, the real thing, and Lynette's on there somewhere as well. And particularly wanted to acknowledge my friend Helen Cross, who gave me some of those slides for the fenfluramine data. Thank you very much.
Thank you very much, Ingrid. And you are now able to observe the 1.5 meters of distance. We're observing good social distancing here. And I was listening closely to what you were saying. And obviously, there's so much interest. There's a couple things I want to ask you about, Ingrid. We'll start with CBD. You talked about the fact that it is proven in terms of Dravet and LGS. And then that's the proven. And then we get into the unproven. So my first question is, when it comes to the other genetic disorders, genetic epilepsy disorders that are represented here today, are we saying it's a no? Are we saying we don't know yet? Or we're saying sometimes?
I think it's we don't know yet is really the answer. And I was trying to give that message very clearly that we don't know, but we need to know. I think it will work for many of the developmental and epileptic encephalopathies. It's not really a precision medicine. A precision medicine means that you're working at that gene defect and you're trying to make that, to fix that gene defect. And Steve will talk about that. But this isn't a precision medicine in the same way. We've already seen incredibly promising findings. It works in Dravet, it works in Lennox-Gastaut syndrome. Lennox-Gastaut syndrome is due to a whole bunch of genes. So there is a good clue there that it will work more broadly. But there are no trials of it in infantile spasms, for example, or many of the other syndromes that we want to know about. And that's why I'm really keen for the CBD making companies to grasp this opportunity and do it really well.
Yeah, I mean, you definitely called out the fact that you want to see more from some of the ones that you call, I guess I've got this right, nutraceuticals instead of pharmaceuticals. So are you saying there needs to be really kind of greater clarity around what's what when it comes to that?
Absolutely, and I'm worried that families don't appreciate that what they might be getting. Maybe you're getting it from someone down the backyard who's growing it in their back garden, and you don't know what you're giving your child, and you wouldn't do that with a tablet, or we as a doctor wouldn't prescribe something that we don't know what's in it. So that's the risk. I know that it's very good for some patients, and I would say in my patients who've been getting CBD, about 20% it's transformational, and 80% the families say, didn't do much or did nothing or it made them worse. It doesn't usually make them worse, but it usually, 80% they're not excited.
But it's also, isn't it, I mean, it's really interesting the results that you were giving regarding the gel, but it's challenging here in Australia in terms of access, really complicated, and also cost. So, I mean, in a way, do you blame families for going more broadly because it's so darn hard to get this stuff?
Yeah, look, I don't blame anyone. I think you're dealing with an incredibly sick child and your lives revolve around trying to improve their quality of life. I completely get that. But I guess I just wanted to caution you and also to show that what you read in the local newspaper or on Twitter is not science. And one of the things, just to go tangentially for a moment, Australia has done brilliantly with the pandemic because we've listened to the science. Yes. Our politicians, Minister Hunt, Scott Morrison, they've actually listened to the science, and that has made Australia shine amongst the rest of the world. So I think it's very important to listen to the science.
I totally agree, and I think it's interesting. There have been some studies, some preliminary studies related to COVID-19 regarding which countries have done well, and if you were to do an overview, it's those who've listened closely to the scientists have done better than those who've listened to the politicians. Speaking of COVID-19, we do have a question about that, and that question is from Arden's mom in Australia, and she wants to know, Ingrid, about the potential impacts of COVID-19 on children.
Thanks for that question, Arden's mum. The news is actually surprisingly good. And by that I mean I was incredibly worried about COVID-19 for my patients who have seizures triggered by fever, and we know that COVID-19 is associated often with fever, and patients such as those with Dravet syndrome, but also PCDH19, numerous other different genetic developmental and epileptic encephalopathies. And speaking with colleagues in China, in Italy, in America, New York specifically, where they've seen huge numbers of patients, they haven't seen an increase in their patients who have fever susceptibility in terms of seizures. They haven't seen them coming into the emergency department. So one can never be sure, and one also, there will be somebody who will have a bad outcome just by sheer numbers with COVID-19, but it's looking like they're not doing worse.
And that is certainly encouraging. I'm going to come to FenFen in a moment. We have a couple of questions about that. But first, I want to ask about a question from Christian in Brazil. What about WWOX?
Yeah. So WWOX, I do the Australian thing and I call it WOWOX because we always make abbreviations. WWOX is most definitely a developmental and epileptic encephalopathy, and it is often associated with very, very severe epilepsy and profound impairment, very hard for you at home, I'm sure. We do have a little research project on that at the moment, if you'd like to make contact with us about that. One of my PhD students is currently writing a paper about it. But it's, you know, many of these genes are rare. So there are maybe only a handful. You'll start with 5 or 6 patients, and then before you know it, you'll have 10 patients. So we're writing a paper on a gene FBX028 with 10 patients. And yet I'm working with Belgian and Danish colleagues, and we're just about to submit a paper with 80 patients.
Well, and I'm struck by the fact that, and I hope I've got your name right, I've got Christian in Brazil, but if Christian wants to get in touch with you, Ingrid, how does she go about doing that?
Thank you. It's that last slide, which will be up online eventually, but it's epilepsy-austin, AUSTIN, at unimelb.edu.au.
I don't know if we're able to put that slide up, but if you can put that slide up one more time just so that people can get that, and then we can come back to, even as that slide is going up, I'm going to ask you another question, and this relates to FenFen. We had a couple of questions around that. We'll try and get that slide for you as we continue to ask the questions. FenFen is definitely of a lot of interest to people, and one of the aspects of this that I found so important is that you said that FenFen was pulled from the market because there you go. So are we looking for,
yeah,
yes, there you see.
There
is the address, epilepsy-austin at unimelb.edu.au. There it is at the end, just in case you want to get in touch with Ingrid, either regarding WOWOX or WWOX or any of the other genetic epilepsies. And also just going back to the natural history study that you mentioned, if you want to participate in that, it's going to start here in Australia, but it is going to go broader, I have no doubt. So if you want to be participating in that, there's the email address for that. So thanks very much for putting that up. And now returning to FenFen before we go on, we won't keep you forever, but FenFen's interesting because you mentioned that it was originally pulled from the market because of implications on the heart. So what we're asked by Paula in Canada, are there any signs of cardiac issues in the FenFen study for this population?
Yeah, very important question, and I think it speaks to my discussion regarding side effects. The company looked very carefully for that. These children had lots of heart echocardiograms to see if there was any cardiac valvular thickening, and there was not, which is super exciting. And I think the reason is the doses were much lower than they used for weight loss in the late 90s.
Are you seeing this with a number of other drugs as well, with things being repurposed for these rare diseases and disorders?
I think, you know, it's early days, and we see that repurposing doesn't always work, like chloroquine. But I think one does need to think about them for each disorder and look at their doses and look very carefully for side effects. So yes, you can look, and this was a nice example of the doses being too high, bring the dose down, no cardiovascular problems
All right.
yet.
And one more question on FenFen, which is, when is it going to be more widely available in Australia? And that's a question a lot for Australia.
Yes, it is.
Any answer?
It's interesting, actually. In many of the drugs we're earlier, like, for example, we had clobazam only 20 years before the US. But the fenfluramine, yes. So, it's not always. We do loads of drug trials in Australia, and we're often early people to take drugs up, because the drug trials mean we know how to use a drug and we're very keen to use it. But I think fenfluramine will be, it's beginning to be rolled out in the US and Europe, and I think it'll be rolled out here, but my whisperer says it'll be a few years, which is really disappointing.
Is there any way people
can
I'm already gonna ask this on behalf of those people who are interested. How do they get that sooner? What do they need to do? Because that seems a long time to wait if people still
it does. I think politicians, I don't know, or go to the company, but I don't know how to make that happen. I'm sorry.
All right. Well, we'll continue to look into that. All right. Ingrid, those have been wonderful answers. Great presentation. And we're going to look forward to going to Steve Petrou in a moment. But first, thank you again. And now we're going to hear from another parent. We talked about the fact that there's so many different disorders that are represented with us today. And we're now going to hear from Renee Swannack, who's a KCNA2 parent, and hear about her journey and what this has meant to her.
Hi everyone, my name is Renee Swannack, and it is an absolute honour to be presenting for GETA 2020. Seeing I'm in my dining room, things look a little different to last year, but please feel free to pause here, go grab yourself a cup of coffee, and let me tell you a little about myself. I'm Damien's mum. Damien is a sweet, happy, loving 3-year-old boy who adores snuggles, spending time with friends and family, laughing at cheeky jokes, watching Bluey and Sesame Street, and listening to classic rock. Damien also happens to have a mutation on his KCNA2 gene, leading to KCNA2 epileptic encephalopathy. Damien is unable to walk or talk, has visual and hearing impairments, and suffers from regular seizures, which I'm happy to say we have under good control at the moment. I'm also the co-founder of the KCNA2 Epilepsy Alliance and one of the co-founders of the medically complex / special needs parenting blog, How to Make Lemonade. Let me tell you our story. Damien was born with what doctors diagnosed at the time as the movement disorder hypereplexia. Doctors had touched upon epilepsy, but were unable to diagnose via EEG due to Damien's movement disorder being so extreme. It wasn't until Damien was 3.5 months old that he began having infantile spasms and was diagnosed with West syndrome. I remember the day like it was yesterday. Damien's results were back from his first EEG. The neurologist on ward rounds handed me an A4 printout on West syndrome with statistics and studies in black and white pictures of babies with EEG leads attached to their heads. And of course the heartbreaking story of Dr. West and his child. Reading that broke my heart. Here I was with my beautiful 3-month-old baby staring down the barrel of an epilepsy diagnosis. Seeing Damien already had neurological issues and was missing milestones, I knew he fell within the statistics of the kids that didn't do so well. The handouts mentioned other forms of epilepsy that children with infantile spasms often progress to, like Dravet, for example. Naturally, that led me to Google all of the above and only found more heartbreaking statistics. My husband and I felt so alone. Who do you turn to for advice and support about something you've never even heard of? Feeling lost and alone, I joined some epilepsy parent Facebook groups, which then led me to parenting blogs and to articles and to stories, and to the beauty, love and strength of children with epilepsy and the perseverance and resilience of their parents. Those were the bits the handouts didn't discuss. 9 months later, we received Damien's official KCNA2 diagnosis. Before we reached the car and found the KCNA2 parents group with other kids like Damien and parents like me, I trawled every single post on the page, every photo, comment, and any link that would help Damien for a glimpse into what Damien's future might look like. And most importantly, I was able to ask questions and seek information specific to Damien for the first time ever. It was on this page that I found my beautiful friend and the only other Australian KCNA2 mum that we know of, Christine Walker. Chris encouraged me to attend GETA last year, which was also where the KCNA2 Epilepsy Alliance was born. We were both so enthused and motivated after last year's conference that we dove in and established the Alliance and in a way built our own little KCNA2 lighthouse in the form of a Facebook page and website, kcna2.com, where we share KCNA2 family stories, epilepsy information, we do myth busting, share links and studies, and importantly, we light a light to our Facebook group of parents. By now I was starting to get comfortable with telling Damien's story. So in order to improve the website and page, I attended a writing class where I met 2 other amazing mothers of medically complex kids. And from there, the Facebook page How to Make Lemonade was established, where we share our experiences, we encourage others to share theirs with us and our audience. This week we hit 800 followers, which we're pretty chuffed about. So by now you can probably tell that I love reading and hearing stories. All things epilepsy, rare disease and special needs. I want to soak it all up like a sponge. I want to hear the inspiring stuff, the boring stuff and the nitty gritty. I want to hear the stories that will make me smile and also those that will break my heart and make me appreciate today for today. And I know I'm not alone. Storytelling is one of the most important community building tools we have. We can educate and we can inspire. I'm sure we've all heard someone say, but it's only epilepsy. In the movies, people have seizures and they're okay. They take medication and they're fine. Those people need to hear our stories. The family members who don't quite understand what's going on, they also need our stories. Newly diagnosed parents need our stories. Researchers and scientists need our stories. Doctors and specialists need our stories. Government bodies and lawmakers need to hear our stories. Pharmaceutical companies need to hear our stories. And philanthropists need to hear our stories. Epilepsy is often an invisible disease. By sharing our stories, we make the invisible visible. Storytelling can contribute to inclusion and connection. It builds confidence and brings about change. Our children are more than just a gene mutation. They're more than their diagnosis. They all have a story to tell, and it's a story that deserves to be shouted from the mountaintops. So, how could you tell your story? The beauty of telling the story is that you control the tone and context. If it comes from love, it will be amazing. You don't need to be a writer or a speaker, you just need to be prepared to write or speak. Firstly, you could begin by writing a journal, which you don't actually need to share with anyone. You could just use an app on your phone. Write down your thoughts, get it out of your head. I know sometimes when I'm writing, tears stream down my face. Those were tears that I was carrying around that weren't good for anyone. Social media is great. You can control your audience. You could be totally anonymous, or maybe create a close group of close family and friends and just share hospital stays, photographs, rough patches, updates, the good days. Share on your own Facebook and Instagram page. Create a public page about your child while still maintaining your child's right to privacy, or a page about your child's condition. Don't get too caught up on likes and shares. A lot of people are on social media quietly trawling for information. At How to Make Lemonade, we often get messages from people about particular posts that resonated. These people often aren't active on the page, but they're there and they're following along. Or a closed group for parents and carers of children with the same condition. This one's really close to my heart. The KCNA2 parent group has been an absolute lifesaver for us. The exchange of information and the ability to ask questions is so powerful. We've even compiled a spreadsheet of information full of everything about our children, their gene variants, medications, therapies, surgeries, their specialists taking an interest, all ready to go should a researcher decide to take a look at KCNA2. You could also write posts for charities and fundraising groups to raise awareness. You could create a website full of information or a blog. You could create your own podcast and get me to come along for an interview. You can make submissions to pages like How to Make Lemonade, Source Kids, or The Mighty. News outlets are also great and have a massive audience, especially around important times like Rare Disease Day or World Epilepsy Awareness Day. So please tell your story. Somewhere a parent is being handed an A4 printout and it's not enough. Be that lighthouse and share your light. Let your story become part of someone else's story. And I can't wait to hear all about it. Thank you.
Wow, and thank you, Renee, for sharing your story and for reminding everybody of the importance of their stories. And it occurs to me that we have hundreds of stories right here in our community. All of you who are joining us, you have a story. You have the stories of all of those people who are in your family and friend networks. And Renee has really given us a powerful testament to how you can channel that, harness that, push that forward, spread that word. And it's important because our next speaker is an example of the kind of person who listens to that. Steve Petrou is a scientist, but I know for a fact that he works with families because that is part of what he does. And he's a big believer in the power of families to get the attention of scientists, to harness their interest and their scientific audacity and skill that some of us as parents, simply most of us as parents, don't possess. We have the passion, the knowledge of our individual children, but we may not have the laboratory and the expertise to drill down the way somebody like Steve can. Professor Stephen Petrou is the director of the Florey Institute for Neuroscience and Mental Health and the head of the Florey's Division of Epilepsy. He heads the Laboratory of Ion Channels and Human Disease, which is a multidisciplinary team of researchers that look at the fundamental mechanisms of disease genesis in the central nervous system, namely the brain. So this is very important. And as I mentioned, he has worked with not only parent groups in terms of looking at what they need scientists like him to study, or listening, I should say, but also with industry. And he holds several patents. Recently, we had another expert come to one of our Get It conferences, and he said to me quietly afterwards, you recognize that Melbourne is really a center of genetic epilepsy research. And you've heard from several of the people today who are a reason why that is the case, and you're about to hear from one more. We want to know about gene therapy. We want to know about how close we are, because we know that medicines are not enough. So it is my pleasure to welcome now Professor Steve Petrou. Steve, good to see you.
Oh, hello, everybody, and thank you, Sarah, for welcoming me to this event. I think I've been at almost every Getter event. This is the first time I've been sitting in my office doing it, but I'll see how it goes. So what I'm going to talk about today is how close we are to a gene therapy. And I'll talk a little bit about, reiterate some of the points that have been reiterated by the former speakers, but to try and give a different slant on it as to the impact and how our understanding of genetic architecture, how our understanding exactly of how genes are broken influences and impacts the way we think about delivering a therapy. And probably first and foremost, what's really important is this genetic architecture. We heard a little bit, and if you can think about it in a very simple terms, lots of ways to break a gene. And precisely how that gene is broken impacts our thinking. And when exactly that gene breaks is really important. And I've just shown this little figure up on the top left that gives some, I'll see if I can get a nice, here we go, nice little pointer. It can happen when the reproductive cells are being formed. Egg and sperm mutations can arise. It's a very infrequent event. We have lots of proofreading mechanisms when we produce reproductive cells, but sometimes that goes awry. And that's actually a natural part of species development. These mutations are critical for driving evolution. Sometimes, though, they also cause problems. But they can also arise at different points during the development. Once the sperm and egg fuse, a whole cascade of events occurs. An egg's fertilized, you become a zygote, then a blastula, and then an embryo. At different points along this cycle, a mutation can emerge here, in which point every single cell's got it. We've heard about mosaicism and other disorders. Depending when it occurs in this process, the mutation might be constrained to one organ or one part of the body, or only a fraction of the cells within the brain. And that's just a different level of disease genesis that has impacts on how we think about therapy. So one of the most common things that we've heard about a lot is new single nucleotide mutation that arises in the child at some point in this cascade. And those mutations can be so-called gain of function, in which point the gene works harder, so it does its normal job, but does it a little too vigorously, or it can even do something completely new that it didn't do before. Both of these things can be the drivers of disease. It can also be a loss of function when it doesn't do as much as it should. Now, what you will find, I noticed in the question someone asked, how can mutations in the same gene give rise to different phenotypes? Well, one of the reasons is mosaicism. One of the reasons can be that depending on the nature of the change, whether it's gain of function, whether it's a brand new function, whether it's loss of function, that also impacts disease progression. Some genes gain or loss both give rise to a disease phenotype. In other cases, only either gain or loss gives rise to the disease phenotype. Even those facts influence the way we think about designing and delivering a therapy. So another common mechanism is changes in the numbers of copies, or the number of copies of a normal gene, deletions or duplications of genes, so-called copy number variations and microdeletions can also break genes and also influence the way. Some of these changes may not be as amenable to some forms of gene therapy and may require different types of thinking. You can actually inherit a copy of an otherwise not so harmful gene from each parent. And recent evidence is suggesting so-called autosomal recessive inheritance, where you have one gene from one parent, the same gene with another mutation from another parent, and those come together and now give rise to a disorder. And that's probably more common than we thought. I've seen 10 to 15% of all cases may arise from this so-called autosomal recessive inheritance. Let's see where the genetics pans out with that. But it's something, again, we need to think of, because in this case, it's two different mutations, one in each gene. That also may influence, if we're thinking about state-of-the-art CRISPR gene repair, we now may have to think very hard about which gene that we would go to repair, etc. Inherited mutations in the X chromosome that impact females. Now, if males are not affected and act as carriers, they can increase the prevalence of that mutation in the population. Again, that drives decision-making around there's going to be more of those patients, and the business side of drug discovery and of pharmaceutical company operations kicks into gear. And these things influence how they make a decision around to go after a particular type of therapeutic modality, whether it be gene therapy or something else. And as Sam alluded to, there's a massive region of the genome that's largely unexplored, the so-called non-coding DNA. And we're just starting to scratch the surface of that. Genetic mosaicism that I mentioned, epigenetics, where you can modify the gene chemically. The body can do that in different ways, and that can also influence disease progression. As I said before, how it's broken determines how it's fixed. And I just want you to just sort of have that in mind when we think about, as we look at this talk going forward, and as I describe some of the findings. This is the cycle. We have a patient, genetic diagnoses are made, we understand about the nature of the mutation, not only the gene, what exactly that mutation does to the gene, whether or not the patients form a spectrum and there's loss of function and gain of function. How do they stratify? Clinically, does that stratification relate to some simple genetic signature, or is there other components, such as Lynette mentioned, around environmental influence? All these things need to be somewhat understood so that we can deliver this so-called targeted therapy. Without all that knowledge of the patient phenotypes, of the genetics, of the consequences of those genetic mutations at various levels of organization in an organism, it's very difficult to think about targeted therapy such as gene therapy. So what even is gene therapy? And it's a bandied about term. It's used a lot. Some people think it's exclusively the delivery of a gene. Other people think it's medicines that influence genes. So I like the broader definition. It's a treatment that directly operates at the genetic level rather than the protein level. What do I mean by that? I mean, protein level, most pharmaceutical agents that we take act on a protein in the body, whether it's an ion channel or an enzyme or some other protein. And proteins are the little machines that are built from the blueprint provided by our genes. But mutations, we know, occur at the genetic level. And as some of the participants have asked, and as this concept has been raised, what about all the other phenotypes that emerge in the patients? And how do we think about fixing all of those? And it's often easier to think about that if we go back to the very root cause of the gene. So gene augmentation therapy can be to deliver a new gene to the body, and you would do that if you know that a gene is broken, not working, and to put a new gene in will replace and restore the function that broken gene cannot support. Then you can use other methods that don't require the complete delivery of a brand new gene, but other approaches that can switch on that gene or another copy or the wild type version of that gene, turn it on, make it work harder. That's possible. In other cases, you might want to turn a gene off. So there's gene inhibition therapy. And again, we use either other little tiny fragments of a gene, or we can even use other medicines to manipulate genes in order to try and counter what we know is the fundamental genetic mechanism. So these are the broad definitions, and I'll give some examples today of how a new gene delivery has been thought about, how we can turn a gene on in a specific way with a gene therapy to both target the same disorder. And just to give you an example and to show that there's actually quite a lot of thinking, quite a lot of hope, and quite a lot of progress in this area. So as I said, what's so good about gene therapy? Well, gene therapy targets the cause. I mean, Ingrid so elegantly explained to us, the current medications, the best way to get out of them, and this need for a magic bullet. So in very simple thinking, if you go right back to the very fundamental cause of a condition and you repair that, all the things that emerge as a result of that change in function should be addressed by fixing the root cause. We don't have to worry about the 12 different things that happen after you break a gene, how it impacts seizures, how it impacts cognition, how it impacts movement, how it might impact gastrointestinal function. We hope that those things can largely be captured by the one approach. And I can say gene therapy has arrived for neurogenetic disorders. Maybe not obviously for every single gene that we're talking about and that we heard about today in our participants. But as Sara so lovely, sort of insightfully told us, that they're going to fall one gene at a time. And that's what's important. And the important thing is, how are those decisions made about which genes to address? And clearly, the work of Ingrid in her natural history studies, the work of patient groups such as GETA, and all those around the world is critical, because if we can understand the patients, we can understand the genetics, we can understand the disease mechanisms. Those elements are all important factors in saying, well, we're going to go after Dravet syndrome, or we're going to go after DNM1, or we're going to go after KCNQ2, as we understand more and more about how we're going to go after it. Probably one of the earlier cases, and I'll talk to you about it now, that how successful gene therapy can be, is the example of spinal muscular atrophy. It's a terrible disease that robs people of physical strength, affecting motor nerves in the spinal cord, taking away the ability to walk, eat, or breathe. Number one genetic cause of death for infants. It's caused by a mutation in this survival motor neuron gene 1, SMN1. And now there are two competing therapies that are out there, and I'll describe both of them. Fundamentally, this SMN1 gene is missing. This is a picture of a bit of DNA. It's gone. It's not doing what it's meant to do. When it's not around, these special nerves or neurons that control our motor function die. They can't work, they degrade, and then all of a sudden, if you can't control your muscles with your nerves, you can't support the function you need. So you really, really, really need this SMN protein. The gene SMN1 makes SMN protein that keeps these neurons alive. So it's quite a simple thing to think about it. Well, if we had SMN protein, we keep the neuron alive, we should address the disorder. Now, in humans, there is an SMN1 gene. This is the one that's missing in the disorder, but there happens to be this very closely related gene, SMN2. And it sits there, and it's not as critical to function as SMN1. And genetically, it's altered in a way that I'll talk about in a second, that makes it less active through a small little genetic change. And if you can use a drug called Spinraza, and you can activate this SMN2 gene by reversing that small genetic change in it, you can then produce SMN protein. How does that happen? Well, this is how we look at a gene. These little bits are the building blocks that come together to make the little SMN protein. The bits, the lines in between are removed, and you string these all together like a string of pearls, and they come together and make a protein. SMN2 has this number 7 piece in it. This number 7 piece is critical to have normal function. Normally, the number 7 is removed. If you can increase the amount of this number 7 in SMN2 that's removed and it doesn't do much, it then makes a full-length SMN protein. So this tiny little piece of DNA, an artificial DNA called Spinraza, was made, and it attaches to the DNA here, and it forces the inclusion of this number 7 piece. And by forcing the inclusion, you make a different version of SMN2 that makes a full length of the SMN protein. And this is called Spinraza, and you may have heard about it, how it's transformed the lives of these children that have been wheelchair bound and almost certain to die young, have now getting out of their wheelchairs, and they're walking and they're living productive lives. So, and this is a form of gene therapy that doesn't require replacement of the whole gene, but it was produced because of our detailed knowledge, not only of the disease gene SMN1, but because of our detailed knowledge of a partner gene called SMN2 that didn't do so much. But the fact that it was there triggered thinking around, well, how can we activate this sleeper gene to restore function? And then that's how information that's maybe a little bit peripheral can be used in very inventive ways. There's also another approach. There's a new drug called Zolgensma that's close to being released. I think it was recently approved in the European market. And the idea here is, okay, let's not bother with SMN2. Let's get a full copy of the SMN1 protein, classic gene therapy, if you will. And that means somehow you've got a package, you've got to get the full length of the SMN gene, you've got to put it into some system, it's called a vector, so that when you give it to a person, it actually gets into the neurons of interest. And that's the job of the vector. In order to do that, a very common vectors used in humans are we take advantage of natural viruses. And this virus is called adeno-associated virus, and it often it causes some respiratory and other disorders. And you can see here, this is the virus with all its little bits. Now, we don't need a lot of the functions of the virus. We only need the bits of the virus that help us get genes into cells. We don't need all the parts of the virus that are used to do other things. So through genetic engineering methods, you strip away all the other parts of the virus, and you're only left with the part of the virus called the vector, which is the bit that can carry the DNA. And so this now won't cause the same disease that AAV9 causes, but it does hijack the important function of this being able to carry a gene. And you can see in this slide that we now have the SMN gene carried in the AAV vector, ready to be delivered now to patients. The good thing about the way you engineer this gene, the new gene that's put in doesn't become part of our genome. It sits peripheral to that, and it's better that it doesn't integrate because that can cause its own problems. So we can genetically engineer it so that it stays separate to our genome, but sits in the cell making SMN protein. And when it does that, neurons are happy. And there's been some wonderful results coming out of trials with this, and we'll soon see, hopefully there's been some hiccups recently around the development of this, but hopefully those hiccups will be resolved and we'll see the broader adoption of this. And this is just one story of a little girl who was on the Spinraza treatment. You see here, she'd been treated for now I think she's in her late teens or early 20s. And she was one of the first patients on the clinical trial. And if you can read this, she began to notice significant improvements to the health. And importantly, her disease had ceased to progress. She had more energy. Here, and you look here, I think the day I knew I was getting better was when I walked up a flight of stairs in my house for the first time in 2 years. Something I never imagined I would be able to do again, considering that this is a progressive disorder. It gets worse and worse over time as more and more neurons die. And one of the other important things is that the time it takes for the effects to be reversed. This little girl took years to not only halt the progression, but then for positive signs to be seen. What's on the horizon that's maybe more relevant to the participants in GETA today? Well, gene therapy for Dravet syndrome. There's two that we know. I'll talk about these two first. This is a company in Texas called OPCO that have a technology called AntagoNATs. And this, again, is a way of waking up the SCN1A gene and making the healthy copy be produced at a higher level. And this has shown very good results in preclinical models. They presented some data at the American Epilepsy meeting last year, where they're going to increase, the development, and we hopefully will see clinical trials emerging soon. Another technology by Stoke Therapeutic. Both of these really do rely on antisense oligos, small artificial pieces of DNA. Stoke have got another method for activating the SCN1A gene. So it's using the natural gene that's there. It's doing a slight genetic manipulation of it, but it's now making way more of it, and to counter the loss that is the common mechanism in Dravet, as both Ingrid and Lynette told us, that loss of this gene is what causes the large majority of Dravet cases. These things restore that loss by making more. There's also other work, and the companies haven't fully been disclosed. There's work happening around delivering a full copy of the SCN1A gene, as I mentioned for the SMN1, using, in this case, a non-AAV method, in another one using 2 AAVs. One of the limitations of gene therapy is, I mentioned those vectors. How big a gene can you put in a vector? And sometimes some of the genes that are broken in epilepsy are too big to put in a single vector. And you can see here in AAV, the SCN1A had to be split in a certain way that lets us get around that shortcoming. What else? I talked about, we just recall this thing, how it's broken determines how it's fixed. Gene therapy targets the cause. What's so good about it? Well, what's so good about it? It's more than just seizures. And I know that question was asked earlier. And if you look at it, this is a particular form of gene therapy around an antisense oligo. I won't disclose the actual disorder here. But what you can see is that you can look pre and post. This is an untreated animal model. This is an animal model. You can see the number of seizures, 20. Following the control treatment, it was having 30, 40, 50 seizures every 3 days. Now, if you can look at this animal, pre-treatment with the drug, post-treatment, no seizures. Pre-treatment with the drug, post-treatment, no seizures. Pre-treatment, post-treatment, no seizures. So this drug could abolish the seizures. The animals without the drug died after 100 days. The control treatment didn't help. The animals that were treated, one dose, remember, that lasts many months. And what you're seeing here is the drug wearing off, not the animals getting used to the drug. But as the drug wears off, they start to die again. But you can see how long-lived the response can be. These drugs can be delivered every few months in the same way that Spinraza can be. Remarkable effect on survival. Epilepsy goes away, animals no longer dying. There's this other test we do to essentially say, how well do the animals think? Well, without the drug, with the disease by themselves, they don't think so well. With control drug, doesn't improve that. As soon as we treat them, their thinking goes up to the same level as a control animal. There's a behavioral response test we do. You can look here, untreated animals, control-treated animals, animals that are treated with the drug, thinking as well as a normal animal. Huge improvement. And this is just showing you this data to say, going back to this point about treating root causes can have ramifications through multiple levels of the phenotype. And that's really the great hope for gene therapy. All of these programs I've shown you so far are at some stage in clinical development for children. This is an earlier stage work that came out in rodents for DNM1 gene, and they were here delivering an entire copy of the gene to the mouse model of this fitful mouse with these mutations in DNM1. Soon as they gave it a complete replacement gene, they got wonderful improved growth relative to the animal that wasn't treated, improved grip strength, ataxias went away, fewer seizures. The animals below were just given the control treatment, and they had all these problems that were there without any impact. You can see the number of degenerating neurons. Lots of the phenotypes were not impacted by the control treatment. All of them were impacted in a very positive way by gene replacement. This is at an earlier stage, but companies are thinking about treating dynamic mutations as well. So I know many of these genes are on the minds of participants in this symposium, and rest assured, one by one, each of them will be done. It's just a matter of timing, a matter of resources, and the matter of the number of people globally, the number of scientists available to be working on each of these, and then the number of companies that are able to take the advances made by the science and the genetics and the clinical work to package it all together and develop it into a program. You look at the scorecard of a company like Ionis, just how their technology, these aren't all rare disorders, but they're just disorders where their gene therapy technology has been applied. You can see one here, DNM2. They've got a collaboration with a French company called Dynacure, but they're interested in applying their technology to a whole bunch of different disorders. Other programs, just giving you a, Orchid Therapeutics have got this other gene therapy for this leukodystrophy. Biogen is interested in a ALS gene therapy. Roche is looking at an oligo to destroy faulty Huntington protein, Huntington's disease. So there is a lot of activity. Every success that occurs in each of these really prompts companies and scientists and clinicians to look at the next thing and the next thing. So we wish all of these programs great success so that there is a great positivity around what gene therapy can deliver. We've all heard about CRISPR. This is my last slide. We've all heard about CRISPR as a game changer, and this really shows it beautifully. CRISPR has the ability to take out a point mutation and put the corrected version in. That's the promise of CRISPR. Huge technical challenges ahead of it about how do you do that to every neuron in the body. And as I said at the beginning, not every single genetic change is the result of a point mutation. Many are, but not all of them. So CRISPR may never be the universal panacea. It's just going to be one aspect of an arsenal of different therapeutic approaches that will be able to be given to people. And this is probably still needs more and more development to be able to treat the sort of conditions that we're dealing with now. So before I end, just want to acknowledge my own group up here. I'm having a meeting, lab meeting, and also Sam and Ingrid, who have been obviously the beginning of this journey. They started the journey by discovering the genes. We work very hard to think about therapies, disease mechanisms. We reconnect at the end once therapies are provided. Then we hope to reconnect and look at the way these therapies impact our patients. So thank you very much.
Thank you so much, Steve. If they are our co-alphas, we're not quite at omega, but we've made a lot of progress. And certainly you've been telling us about what some of that progress is. And it is exciting. And I think it's one thing I wanted to mention, my friend Kelly Cloesy, who's a member of GETA, she's not here in the office with us today, but she made a point sending me a text about, for families, the significance of where we are now. And if you're just starting the journey, or if you're one of our families where your child is 25 years old, we're making progress. And there's a lot of good things that are happening. And I think that's one aspect of what makes your research profound and important. My first question for you is, do you feel that sense of excitement that we as families feel about the potential of cracking these things open?
I mean, I have the emotional connection when I see the patients talk to the parents, and that's a big motivator for me. But then when dealing with the delivery and the discovery of it, you get immersed in the myriad of problem-solving steps you need to do. So doesn't feel profound at all. It just feels like a long string of things that have to happen and a lot of things that have to align. And even at the end of it, because of that, it's if you work backstage at Disneyland, you don't get the same effect.
Well, I'm really glad because I can see front of house. And since I can't do back of house, so it's probably a really good place for me to be. But we hope we can also provide the cheering section because we'd like to believe that that is important. I do have a couple of questions for you that relate directly to your research. One of them is, do you have any view of whether gene therapy for children with genetic epilepsies could have positive outcomes like SMA, where you could do gene therapy later in life? Or are you in particular looking at things that would be in utero? What's your thought on that?
So, I mean, that is really one of the central questions that's asked at the sort of beginning of any program. And it's Spinraza. Take Spinraza as an example. It was originally given to younger kids, and they saw some very positive results. And no one really knew what's going to happen when they give it to older kids. And of course, if you've got a drug and it's safe, why wouldn't you try it in older kids? And when they do try it, it was remarkable success. Now, whether that's going to occur for every single gene, we don't know. And I think I'm always hopeful, and maybe a little bit more hopeful than others, that we can turn back a lot of that pathology. But I think everyone's thinking about that. We're always going to try, and we're never going to know really until the clinical trial is done.
All righty. Can ASOs be used for nonsense mutations? That's a question from Marta in the US.
ASOs, yes, they can. And the examples I gave in Dravet syndrome, both of those examples are used in nonsense, where the ASO acts on the healthy copy, because bearing in mind for these de novo disorders, you've got one healthy copy, one copy with the mutation. So we can act on the healthy copy, increase its workload to make up for the loss of function in the other.
All right. Well, that's certainly optimistic. Any information on SCN8A? That question is from Diego in Brazil.
Yes. So SCN8A, the encephalopathy is driven by a gain of function. I think that's fairly secure, our knowledge around that. There are SCNA mutations that give rise to loss of function, and that results in a less severe form of epilepsy called absence. But the very severe gain of functions one produces the very severe disorder. Now, I know that there was a recent paper that came out by Miriam Meisler looking at an ASO for SCN8A. One of the concerns there is that overdosing that drug could cause death because 8A is so important. So there needs to be potentially a slightly different and tailored approach to balance safety versus efficacy. Some genes are a little more on that edge, the razor's edge, in terms of the amount of efficacy you need versus going too far. I know there are companies called Xenon in Canada that is producing a drug that's specific for 8A. They're in various stages of development, and let's hope that that's not a gene therapy, but that's a drug that specifically targets the aberrant SCNA protein. So there is some hope that their drug will work.
You know, actually, you raise a really interesting question, Steve, because I know one aspect of your work, you have the work where you're there at the Florey, and then you also have your work where you do work with corporations, and I know you've got your patents. What's the importance in your view of straddling that divide and connecting academia and pharma, big pharma, and little pharma?
Because, I mean, having seen how pharma operate and make decisions, I've taken a view that, can I make these programs as attractive as I possibly can so that they want to start to work on them? And that involves a lot of de-risking activity. So we develop the understanding, can we do the proof of concept studies? Can we develop some of the assets that are required such that a company may be enticed to invest the massive amount of money We think grant money is expensive, but these companies can cost tens or hundreds of millions of dollars to take these projects forward. So can we de-risk it sufficiently with a complete package of information? And that's why I've started a couple of companies to exemplify that process, and I work with companies to try and say, look, these things are primed and ready to go. You know, what's it gonna take to go to the next step? What's it gonna take for you to fully engage? And that's where we think about everything. We think holistically about patient groups, as I mentioned before. We think about the clinical stratification. You asked the question, if I had the drug in my hand today, which group of patients would we give it to? You know, it's not a trivial question.
Not at all. And it's one with ethical implications as well.
Correct. So that's sort of the way I think about it, at least. Wrong or right, but that's my philosophy.
Okay, so let me ask you a question about that, because there would be people out here listening to us now. Some of them would be, you know, with groups that have maybe a large number of numbers, others not so many or not so many yet. If people want to, so to speak, get on your dance card or on the dance card of people who are moving the ball forward, how do they do that? What's your best advice for parents, either with individual genetic mutations or collectively, like us here at GETA? You've been working with us, as you pointed out, for a long time, to get companies to help solve the genetic mutations that are changing children's and grown-ups' lives.
I think aggregating is really important. If they can reach out, and it's relatively straightforward today through Facebook and Google, to find all the patients you can, try to, you know, get your local, you know, national level and international level organizations come together to show the companies, well, there are 500 of us, there are 1,000 of us. Those things matter, because in this space, no one really knows how many cases there really are. You know, and once you start to get a taste for that, you can get a better idea of what level of investment a company might make. So that's important.
All right, I have one more question for you that you touched on, but I just am going to try to nail you down a little bit on the timeline. What are the timelines for clinical trial of ASO in SCN2A? Day, month, year, please.
It's, I mean, you know, I can say it's in the year to 2.5 years window.
That's good. That's soon-ish.
It's yes. It's sort of, you know, it's near horizon. I can't, it's much closer. It's close. It's close. It's going to happen. But there's a lot of things that have to occur before you can put a drug in a person. But I think there's a huge amount of will, and a lot of things have to go right.
Sure.
As well. You know, there's a lot of steps that may not look right, and then that can delay and that can put challenges onto a program.
And just before I let you go, are there any other genetic mutations that you think are getting close, that you would put kind of in, if you would, like if it's a cab rank, what are the other ones that are in the queue?
SCN1A, clearly. I think there are numerous companies. That's probably going to be the first to fall, I think. As I said, SCN8A with Xenon. Maybe KCNT1 is one that's got some progress. That's, you know, that I know companies are working on it, and they've declared their interest on it publicly. So they're some of the ones that I think are going to come early, and we'll see some real progress on. And there are others that I really can't disclose, because then I can't work with the companies again.
And we do understand that. All right. Well, Steve, thank you very much. We appreciate your insight into gene therapies. And I think that for families, there's a lot to think about, and there's reason for optimism in terms of genetic therapies, in terms of medicines, in terms of the future of being able to treat genetic epilepsies in a more comprehensive way. Thank you so much for your time.
Thank you, Sarah.
And before we finish, we have one more speaker, and then we'll have some kind of wrapping up thoughts. We have another presentation by Dr. Katherine Howell, and she works at the Murdoch Institute. She also is their team leader there in terms of epilepsy. One of our GETA members, Chris Pierce, put together a roundtable recently, a really important and effective opportunity to get a lot of people together. Katherine Howell was part of that. Let's have a listen to what Katherine has to say.
Good morning, everyone. My name is Dr. Katherine Howell. I'm a paediatric neurologist at the Royal Children's Hospital in Melbourne, specialising in severe and early life epilepsies. There have been a number of advances in recent years for children with genetic epilepsies. Most notably, the increased use of genetic testing in clinical practice has meant that many more children are receiving a diagnosis, and often much earlier after their symptoms begin than they would have in years gone by. However, there are also many aspects of the care and management of children with these conditions that we still don't do as well as we would like, and which have changed very little over time. We as clinicians, and obviously you as parents, would like to see this change so that all children receive the best possible care and that their families are supported as well as possible through the difficult journeys they face. To make the most effective improvements, we need to do this in collaboration with you. We need to ensure that we focus on things that will make the biggest difference and things which are areas of priority for families. In partnership with GETA, we held a roundtable event in November last year to discuss these issues. The event was run by me, Chris Pearce, and Dr. Stephanie Best, an expert in implementing change in clinical practice. We had over 20 participants, including parents of children with genetic epilepsies, some of whom head up GETA and other support groups, neurologists, geneticists, genetic counsellors, and members of the Epilepsy Foundation. We discussed many things, including epilepsy diagnosis and treatment, management of other important aspects of these conditions, including development, behaviour, and other comorbidities, access to the healthcare system, overall coordination of care, and support of families. We discuss what is currently done well, what are the areas that need improving, and of these, what are the most pressing needs. The group identified a number of major problems and gaps in care. Major issues included poor coordination of care and frustrating and exhausting difficulties in navigating the health system, meaning that many aspects of the child's management are delayed or not addressed. The group identified that this important deficiency is in stark contrast to what occurs in some other severe conditions with complex needs, such as childhood cancers, where care coordination and support are readily available. We want any changes in practice to be able to be implemented nationally. Thus, it is important that people from all around the country who are affected by these conditions get to have a say in what are the areas of greatest need for them. Our participant group contained only people from Melbourne and Sydney, and with lived experience of only a small number of the many different genetic epilepsies. So, we need more information, and for this, we need your help. We are using the findings from the roundtable event to develop a questionnaire asking about the experiences that you and your child have had with the healthcare system. This will be open to all Australian parents of children with genetic epilepsies and to clinicians. We'll ask you about your experiences of the healthcare system. We want to know which resources were available to you, which you utilised and what your experiences were with them, what worked and what didn't, what were the barriers to and enablers of good care, what could be improved on, and what you think are the most important areas to improve. We will use the findings of the study to develop short, medium and long-term goals to improve care of children with genetic epilepsies and their families. The questionnaire is not quite ready yet, but when it is, we would love it if you would share your views and experiences with us. We plan to advertise it through GETA, so keep an eye out for more about this in time. Thank you very much.
Thank you so much, Katherine. We appreciate that, and we look forward to seeing the results of that survey, which is very important. Speaking of surveys, our Survey Monkey is completed. 43% of you joined us from the state of Victoria, our home turf here in Australia, and 20% from the United States. So we really have a significant number. My understanding is that there are more joining us from the United States than from New South Wales. Wow. Anyway, we have a lot of people in the United States, so that might be part of it, but nevertheless, we appreciate it. We very much want to thank AIA Australia and the Epilepsy Foundation for sponsoring our GETA conference. We expected to be live and in person and, give you some chocolates. So instead, we're going to have to give you your bonbon verbally and say how much we appreciated the fact that you were able to sponsor us on Zoom this year. We hope to be back in person next year, but we also hope to be able to continue this ability to speak remotely to those of you who are interested in genetic epilepsy research, whether you're asking us questions from Brazil or the United States or Turkey or Yemen or wherever you're joining us from. I want to close by saying a huge thank you to my GETA co-founders, Danielle and Danny Williams, Chris Pierce and David Cunnington. I'd like to thank all of our speakers who are here today. And I want to thank all of you for making time to join us and from wherever you joined us today. The final thing I want to note is that each one of you has been told at some time or another that what your child has is rare. And in the community of rare diseases and disorders, that often feels like a very lonely place to be. But there are two things to remember about that. The first one is that rare has two definitions. In rare disease, it's not always something that you want to hear. But it's important to remember that rare is also precious. Living here as I do in Australia, I'm reminded of pink diamonds. They're the rarest of the rare. 90% of pink diamonds are mined here in Australia, but of the big mine, Argyle, in Western Australia that mines them, they make up only 1% of the diamonds that come out of that mine. They're rare, and so are our children. If you look on a website and you read about rare diamonds, you'll find that they're harder to cut, they're rougher, they're more complicated. And I think that's true with our children. You'll discover that they're more precious and that they're very beautiful. And I think that's true with our children too. So I think we need to remember that rare is difficult, but it's also very, very special and precious. And the final thing that I want to leave you with today is that we are rare, but we're not. Because the reality is that we're not just our individual genetic mutations. We're part of a much broader community, a community that spans the globe, a community that makes us one of that 1 in 26 number that we mentioned before. And that's where I'd like to close, because that's not very rare at all. 1 in 26 people around the world will be diagnosed at some point in their life with epilepsy. That means that it's a significant number. We just heard from our last speaker about the importance of those numbers in creating a community, in talking back to stigma, in reaching out to the experts who are going to cure this, and for making people realize that it's important. It's important for every one of your children, and we have the right and the need to ask for, indeed to demand, the help that we need for a large population of individually rare children that has been overlooked and is precious. Thank you.